Right, as I told you previous slides, so I can classify two sections. The first section is bio applications. So we keep trying to develop portable sensing techniques through the LSPR or source or LFA techniques. So all of the we can call them otherwise known as the portable on-site detection tool. And other things we are working out the synthesis of nanomaterial to effectively deliver chemical to raw material to the to kill the cancer cell. And also we simultaneously we want to put in one of the another function to obtain the imaging data. So we want to try to put in two functions. One thing is to effectively deliver to the materials. Other function to obtain a more screen cancers. And also we need to decide is some the margin of the surgical margins the when we found some the specific disease or cancer areas. All right, other thing in the material sections, due to time limitation, I'm gonna skip this. For later, if I have a chance, the more time, and I will introduce the some material section, how to synthesize a functional material to give it more function for give it enhance the some biocompatibility or improve enhance the energy capacitance to couple with the implantable energy devices. Other thing. Okay. You can look at some publication in terms of applications and the material sections. All right. Now I want to go to move to the today the title portable sensing technique for the harmful detection technique from the food, especially the infant formula sample. So nowadays we live in a globalized wave of culture and foods. So we can easily find many different kinds of food. So while many food-related incidents are constantly increasing, the safeguard is needed to protect our health and the safety from harmful substances in food. The safeguard can be divided into three categories: the biosensing or adult materials, bioimaging sections, and the for a safeguard, the can be divided into three steps according to the level of copings. The first step is to monitor and detect the target materials improved. So in terms of the info sections, so that's why we need this promising sensing technique. So today in my talks, I want to focus on portable on-site detection technique for the harmful substances and also a specific disease. And the adjunct material, adjunct section means your technique related to removal and treatments. So I want to give a simple questions. What should we protect from? So how we can do? In other words, so what makes the food deteriorate? So we have to consider the following three steps, three groups. So how to detect the harmful substances. So when you are in the market or in your field. So that's why we need the portable and the point-carrier techniques for the monitoring and detection of harmful substances in on your site, and also how to effectively remove and purify the contaminants and the harmful substances from your in our body or from our around of you, how we can do. So later, it seems like related with the materials, functional materials. We need to design and synthesis of functional material to selectively remove the harmful substances and some specific diseases. And also we have to determine the more suitable and appropriate for the surgical section. And also we have to verify the location of the harmful substances in our body or some around of you. So that's why we need the bioimaging process, and it's rightly related with the biomedical instruments. All right. This slide shows the chemical contaminants in the food. It's one of the food for a cycle. This slide shows the one of the example. We can imagine that there is the one the nuclear plants. So a few years ago in Japan, there was accident, the nuclear plant accidents. So I think about the older world, some they are partially contaminated from the radioactive nuclide materials. So and also, we can sometimes we are suffering from some nuclear the contamination in our body. So and also that makes contaminations in food and the fishy others all around the you. So that's why one of the food chain. So so we can think about it even at this moment, which you are not aware of. So we are clearly seeing more opportunity to be exposed to various environmental pollutants. So I just give us one of message. So nowadays, so we are not safe from the all around the you. You're drinking water, drinking eating fishy and food. So we have some they think about how to keep right the protection of our body or the life from some contaminations, other the nuclides and the harmful substances. So this slide shows that how to figure out how to monitor harmful substances in the around WU. So how we can find the safe food and effectively keep our the health. So if there is a sensor technology that allows ordinary people to easily find toxic substances, so while looking at the market, so it will be one of the solutions we are looking for. So we can think about it, how to figure out which sample is safe and right. So there's right if there's no any contamination food or not. So it's more easier to find out and figure out to you can choose the safe food on your market. So that's why we need portable sensing techniques. So you can look at it. There's a food probe. So that is the level of dilutions, counterfeiting, and the mislabeling food products. That is we can call that food frauds. So that's otherwise we say they're fake foods. So we can simply classify three categories. As I told you previous slides, we can sensing part and the monitoring and hazardous sample. Some due to time limitation, I will the simple skip it. And this slide, I want to compare with the general detection technique and portable sensing technique. There are conventional technology that can detect harmful substances in foods. And these techniques, such as HPLC and LC-MS and others, the conventional sensing techniques, so have the more advantage though. And have the disadvantage of requiring a typical analysis time of 1 day or longer per week. Despite the sensitive detection, sensitivity and complicated sample processing. So in this study, I want to focus on developing simple sensor technique that can be used in the field, so while maintaining the highest possible sensitivity. So you can look at it, what's the comparative table and in the general detection technique and portable sensing techniques. So I want to combine also both of advantages, both of benefit. For the general detection technique, give you some more sensitivity, but it takes so much of time though, so we can obtain more highly sensitive signal. And a portable sensing technique is more simple, so just requires a small amount of sample requires, and so it takes the right, a few minutes. So within 10 minutes, around 10 minutes. So that is the usually it takes a time to obtain the signal, so to monitor your the food and your in your body, some the food is specific disease and harmful substances. And so what this slide shows you what is the portable and the on-site detection technologies available. So this slide shows that. So first thing, the localized surface plasmon resonance, otherwise known as LSPR, and the source and surface enhanced Raman scattering, and LFIA. So simply we call that lateral flow immunoassay. You can imagine that to pregnant testing. So you can just drop it on your urine. So we can simply figure out if you're pregnant or not by discriminating the color changes with your naked eye. The 5 minutes, 10 minutes, that's enough. So that is one of the portable and on-site detection technique. And the colorimetric changes. So that is used one of the tool commonly used to monitor harmful substances for on-site detection is possible. All right, this slide shows what is the advantage for the portable sensing technique, portable sense techniques. So we got us some, we can simply imagine that for the point of cares, portable sense technique have the several advantages. So simple and rapid, the selectivity, and the very cheap, and the portable, that's the available. So what are the key decision criteria for the portable sensor developments? You should be able to finish the analysis within a short time, at most 1 hour. So the most recommended time is within 10 minutes. And also it's hard to say that it is a portable sensors when it's longer and the 30 minutes and the 1 hour. All right, it's a little bit difficult that this is the portable and on-site detection technique. So recommending time is the less than 10 minutes. So in our work, so we are also focused on reduce the detection time. So within 10 minutes. And it should be easy to see and follow the protocol at the middle school level and or the high school level. So right, just the protocol and all of the sensing procedures is very simple. So to follow the using a portable sensing technique, so we can introduce our sensing technique for the ordinary people to handle it and to get us some more useful signal from the your sample, such as the food and also some of your urine flow sample. And usually you should maintain a confidence level of the 80% or more. And also the higher the value, the better. It should be easy to carry around. And the portable, there is more important there. So that is why we call that portable sensing tool, sensing technique, and otherwise point of care techniques. And compared to conventional detection methods, and the advantages of a portable and also point of care technique sensors, so such as or LSPR or LFA or colorimetric changes. So generally rapid and simple and sensitive. That is the all thing is the available. So this slide shows that how we can obtain or the advantage for the portable sensing technique. This slide shows that how to improve portable sensing technique. So how we can make it more promising portable sensing techniques. So we have to consider 4 categories. So one thing to develop and screen bioreceptor material towards specific your target materials. So once deciding your target sample, such as right in this slide, right in this works, we choose the melamine sample. So this is one of the example. Once it's deciding target sample, we have to try to screen and find the more the assurable, most assurable the bioreceptor material. So there are several tools to screen and choose the bioreceptor materials. So in our lab, in my lab, we are working out body display techniques to choose and screen bioreceptor material to the having the highly sensitive and also selective property to the target sample. And the second thing, you can imagine that on-site or the portable sensing technique. So right, there are several steps of sample preparation step or the complicated sample preparation step. So right, that portable sensing technique is not available to obtain useful signals. So when you're at market or so you're in your house, so you are focusing on developing simple sample preparation steps. Just we need just only one filtration or just the mixing. So that's it. So um, so if you need more complicated step, it's not useful to combine a portable sensing technique. So that's why in our industry, so we try to obtain the signal without sample preparation step. So if we need, so we develop and also we make us a simple sample preparation step, just one step or in 1 minute, that's enough to purify sample or separation of the sample from the blood and urine, the food sample. So third step, we have to immobilize and functionalize bioreceptor material onto the for conjugation probe. So how we can design a smart conjugation probe materials. So that is why we synthesize functional nanoparticle. Usually, commonly use the coordinate particle in the area of portable sensing technique. So there are several tools, several protocol, and there are a bunch of papers so regarding the design of coordinate particle. So coordinate particle commonly used in the field of portable sensing technique. There are several advantages, biocompatible, and also it's more easily handle the shape and size, and also we can give a functionality onto the coordinate particle. So in this slide, in my groups, so we can obtain some several shapes of coordinate particle, the hollow shape, a star shape, a cube, and the spheres. So we can fully manipulate the design, the shape, and the size of coordinate particle. So finally, we have to compactably integrate all of function in the one strip. In a single sensing strip, we have to put in several steps. So in this is one of the example to operate the sample preparation from the sample preparation to obtain the signal. So this slide shows the lateral flow assay. So that is previously I told you that that is pregnant sensing strip, exactly the same thing. So in my team, so we can design of lateral flow assay strip devices. So to do that, we can immobilize the coordinate particle and the functional with the receptor materials, and also we can pattern with the test line and the control line. So finally, once introducing the sample, once the dropping sample onto the sample pad, so So, in 5 minutes or a few minutes, so we can discriminate the color changes, color differences. So, if there is the contamination or disease in your sample or not. So simply we can figure out the where contamination or not with your naked eye. So in these days, so we want to obtain the more quantitative and qualitative both of data. So that is why we are coupled with the professional leaders. So later, I will briefly introduce how to combine with the LFA strip devices and the leader devices. This slide shows you what is the localized subproduct redundancy. It's one of the for a principal surface product redundancies. So I think everyone have already known about it. It's a well-known, well established the principle and knowledge of surface product redundancies. So there are noble nanoparticles which are having a bunch of free electrons. So once the instance light coming in onto the cold nanoparticle and noble nanoparticle, depend on size and shapes, so we can triggers the plasmon resonances. There is we can call that surface plasmon polaridon. So by match with the instance light and the cold nanoparticles, the intrinsic the wave frequencies, we can greatly increase substrate plasmon resonances. So through using these phenomena, we can improve and increase the sensitivity, and also we can get some more promising signal from your sample. So, as I told you, so put in terms of portable sensing technique, so definitely there are sensitive, simple, rapid, and cheapest the sensing tool. So that is the advantage for the developing portable sensing technique. This slide shows you how to fabricate the surface plasmonic active substrates. So in the compare with the others have already developed substrate plasmon resonance devices. So in my groups, so we try to transparent plasmonic active substrates. So that's why we choose transparent substrates such as plastic and also glass substrates, and also via surface treatment with the APTES, and by using the amine functional groups, so we can condensely pack the golden particle onto the transparent substrates. So you can look at it. We can verify how to pack with the golden particle onto the substrate. So as I told you, as expected, so we can observe there are a bunch of golden particle condensed packed with the monolayers. So where you can look at this plasma active substrate, and also we assembled with the cubed shapes. So and also we can put in cubed cell. So this is commercially available, and also we commonly use the UVB spectrometers. So simply we can integrate with the cubed cell, and also we can use the to detect your sample by dropping into the cubed cell. So just the 10 minutes, that's enough. We can obtain the signal from your sample. And so after assembling our plasmonic active substrate, so to verify the usefulness of our portable sensor devices, the manufacturers in our lab. So we selected melamine and histamine as a model sample among the harmful substances in foods. So melamine, you know about it in China, 10 or 15 years ago, there's one of the big issues to contaminations, the melamine in the infant formula sample and the milk sample. So usually in food areas, so it's more important that how amount of contents of amine. So it's related with the protein. So that is why they check it out the how amount of amine in the container sample. So that's why the melamine is the cheapest sample, and it's not the use what has helpful, and it's not the helpful for your the body. So to get us some more the economic profit, they put in for the melamine, such as melamine, for cheapest the harmful substances in your food. Anyway, everyone well know about it. Melamine is the harmful to eat in your body. It's not good. And also histamine, or one of the example sample to the monitors the spoil or not in your fishy or the, some food. So one of the biogenic amines, this is the histamine. So it's very useful factors to decide the spoil food or not. So that's why we choose the histamine as a model sample to detection of histamine from the fish on your market or in the field technique. This is good. So in the Korea, food, drug, for administration, and also all of words is commonly cut off the concentration level 2.5 ppm, and also for the histamine sample, so less than 200 ppm level. So that is allowed to they contain in the foods. But over the 200 histamine, their food is spoiled. So don't do that. Don't eat it, and also it's more harmful for your body. So after assembling our devices, so first step, so we fabricated the plasmonic active transparent substrate, and so we immobilize or functionalize the receptor materials. So we were choosing two receptor material. One thing is the chemical receptors, another is the bioreceptor material. So chemical receptors, according to the reference papers, we choose the para Na material, and also in the another is bioreceptor material. We screen the receptors through the positive display technique. So once functionalizing receptor material onto the plasmonic active class substrate, we put in a melamine sample. So without any sample preparation step, just the infant formulas or milk sample, we can put in our devices, and also just where you can stay in 5 minutes. And also we can detect the UV spectrum, conventional UVB spectrometers, so we can figure out spectrum changes. It's nearly simple. And so in our devices, we can obtain the more the ppb level sensitivity. So this slide shows how to prepare a sample. So this is the we prepared infant formulas, and for the making simulated sample, we added the melamines and the known melamine concentrations, and they mix it with the infant formulas and the TW. And also we so once making the different concentration sample, we put in sample into the have already fabricated the cuvette type LSPR sensor techniques. And also 5 minutes reaction and also washing out, we can detect the different plasma signal with the conventional UV-based spectrometers. And also we try to assemble all of things in a portable sensing devices. That's why we bought one of the small UV-based light sources and the cubed cell, a portable cubed cell, and also Ocean Optics. They supply the small portable types of spectrometers. So we assembled all of things, and it's more easier to carry around, and also portable things, and then we can detect the melamine sample through our assembled all of the portable sensing systems. So with the highly sensitive and the 1 ppb level. So and also as the increase of concentration of melamine, so we can observe the differences spectrometers, and by calculating intensity, peak intensity and peak areas depend on your preference. So we can choose the peak shift and peak areas. So we can calculate the melamine concentrations through the standard curve. So we can obtain the sensitive level 0.01 ppb. And also simultaneously, we want to do cross-check our sensing devices. So that's why we try to detect the melamine sample with the source. So by using the functional light coordinated particle, we combine with the combine with the melamine sample. We can monitor, we can verify the melamine with the source technique. So as expected, as the increased concentrations, so we can successfully detect the melamine with the source and Lama spectrometers. And also another is the cross-check technique. So we can apply the RFA technique for the detection of melamines. So we prepare the melamines with the different concentrations from the 0.01 ppb to 2.5 ppm level. So as is shown in the slide, we can successfully detect the melamine sample. So as the increase of concentration of melamine, you can look at the color changes or diminishes. So as the difference in the color changes, we can figure out the food sample milk or infant formula there is containing melamine, such as the harmful substances in your food and the milk powder sample. And also this slide shows that we want to cross-check to evaluate the applicability in the field. Blind tests were carried out using 20 unknown samples from the Korea Food and Drug Administrations. So we can obtain sample from the KFDA, Korea Food and Drug Administrations. So we did blind tests to figure out the concentration of melamine in the from the 20 unknown sample. So as a result of our experiments, so our sensor device showed there's accuracy of more than 85. So 85, and the analysis was completed within 10 minutes. So therefore, our sense device is expected to use as a technology to monitor and identify food harmful substances in the field. So, and also we keep trying to integrate all of components, the portable sensor technologies. And for this slide shows that, so we they are partially integrated all of components, light sources, sample holders, and the detector parts, so we can assemble all of things. And so it's not the small molecule level. So at the DNA level, we try that. Our LSPR sensing technique is the working well or not. So that's why we choose genetically modified crop sample. So we can verify our LSPR-based sensor technique. So how to sensibly detect the small molecules such as DNA level. So how we can obtain the LSPR signals through our developed LSPR sensor techniques, sensor devices. So as shown in this slide, we can successfully detect the genetically modified crop sample, and we can obtain the very low level of concentration GM. And also another sample, we can detect the cell level in the bacterial cell level. So just only small different thing. So we choose the cell receptor material, and also we functionalize receptor onto the plasmon active substrate. So we can successfully detect the Staphylococcus and using the aptamer receptor materials, using our LSPL sensors developed in our groups. So we prepared spike sample, we prepared pork, and we contaminated some of the Staphylococcus, and also we try to detect the bacterial cell from the pork sample through our developed devices, so we can obtain the successful data from our devices. So, and also we tried that and to detect the norovirus sample. So another thing, so from the small molecule to large molecule, from that otherwise from the DNA level to cell level, so we can successfully detect the target sample through our developed LSPR-based devices, and also to cross-check with the other portable sensing techniques and such as source and the RFA sensing devices. So we can obtain, correspond to the similar data. And so our sensing technique, we suggest that one of the promising portable sensing technique for the applicable in the field. So this slide is one of the example to improve sensibility by using the sandwich assay techniques. So due to time limitation, I will briefly explain the slide results. And also we combine with the source and the LSPR data. The source giving us some more the highly sensitive signal, and the LSPR we can obtain the quantitative data. So we want to combine with the source and LSPR both dual functional plasmon active sensing devices. And just a few years ago, we successfully moved our technology to venture company. So our technique, that is we verify our technique for the working out in the near field. So this company in the keep developing plasmon active sensor devices for detection of disease and the harmful substances from the waters, from the food and blood and urine sample. So they keep trying that. So we will give us some comment and advice to modify and develop portable sense technique based on plasmon active substrate. And this slide shows their conclusions. Our portable sense techniques are simple and rapid detections, and for the food and harmful factors and the urine and blood sample. So usually it takes 5 or 10 minutes to obtain the signal, and also very low detection limits, so around the 50 or 10, so ppb level or ppm level, depend on target sample. So in the for the melamine sample, we can guarantee the signal in the 50 ppb level. So we can imagine that in the near futures we can combine with the cell phone and electronics. So we can realize portable point of cares, portable sensing technique to monitor, to verify something in your body or the food. So we can screen what's the safe food, and also you can figure out so how you can so how your health condition, other thing. There are several things. So it's almost just only 10 minutes.
Yes, sir.
I'm gonna keep it, skip it, all of thing. Later,
Okay.
if I have a chance to right, present more time. So I wanna share with the later rules. All right, thank you so much. All right, to introduce my research work to all members. So
Thank you,
this is
sir.
my final presentation. Thank you so much.
Thank you, sir. So I request any participants to have 1 or 2 questions, and so that you can unmute yourself.
Yeah, sure.
A very good morning, sir. Such a fabulous lecture. Thank you so much for your lecture, sir. Sir, may I know the application of the detection of the GM crops? What is the purpose of the detection of by using your chip on the GM crops?
Yeah. So first of all, this objective to figure out the LSTR-based devices is working or not in the level of DNA, the small molecule level. Sometimes the LSPR technique is already difficult to obtain the promising signal. So for the small molecule, so that's how we choose the DNA sample. So just the GM. So nowadays it's one of the big issue to guarantee of safe food. So that's why we design a more the active GM DNA sequence, just a small molecules. There is a 20 mers, just only 20 mers in the less than 20 mers. And also to detect the active DNA proof. So we design of the captured DNA. So we prepared capture DNA and the sample DNA. So we immobilize the capture DNA onto the plasmon active LSPR devices. And also by using the DNA hybridization reactions, so we can successfully detect the GM sample with the very low levels LOD values. So this is just one of the model sample we started to start. So our LSPR sensing technique having a promising tool to monitor harmful substances so at the DNA level, small molecule level.
Thank you, sir. Thank you so much.
Thank you.
Thank you very much, sir. Thank you very much. It's the time. Thank you so much, sir, for giving an elaborate and very informative lecture on sensor technology. You have covered a lot of information on biosecurity for hazardous materials, biosensors, portable sensing method, gold nanoparticles and surface plasmon resonance, fabrication, and some harmful model samples in food, which are very inspirable to all the young faculty and young researchers. And finally, biosensors and rapid detection for food technology. Thank you so much, sir. And we are expecting many more talks from our university. Thank you so much.
All right, thank you. Thank you so much. So I'm really honored to have a time to share with my research work with you. Thank you. Thank you so much.
Thank you, sir. Thank you very much.
Okay. All right, I stopped my share screen.
Okay, sir. Thank you. Thank you very much.
All right, thank you so much. We hope to see you soon. Later.
Sir, I like to thank Professor Basveshwar, sir, for introducing me to our university. Thank you, sir.
All right, thank you.
Thank you.
I started my video and then my voice. Thank you.
Thank you, sir. Now, it's time to invite our today's guest of honor, Professor Vikramaditya G. Yadav Garu, University of British Columbia, Canada. May I know, sir is online?
Yep, I'm here. Good morning to everybody.
Sir, good morning. Welcome, sir. Thank you, sir. Thank you for giving your concern to deliver a talk in our fabulous international webinar. And I request Dr. Jyoti to introduce sir. Over to Jyoti,
Prashanthi.
ma'am. Yes.
Yep. Yeah, speaking. Good
Yes.
morning.
Good morning.
I'm Dr. Jyoti, Department of Chemistry, Mahatma Gandhi University, Nalgonda. It's my privilege to introduce today's speaker, Professor Vikramaditya G. Yadav from Department of Chemical Biological Engineering, School of Biomedical Engineering at University of British Columbia. Professor Yadav is a great scholar working on carbon capture, pharmaceutical manufacturing, medicine, and environmental remediation. His approach is following and learning from Mother Nature and applying these learning in manufacturing processes. His research in medical biotechnology focuses on two critical strategies. One is building tools for discovering new drugs, and other is to identify more efficient and green synthetic way to manufacture new drugs. His multidisciplinary partnership with different research groups, universities, and industries helped to build three-dimensional structured brain organoids from stem cells, a cure for Alzheimer's disease. He did his Bachelor of Applied Sciences in Chemical Engineering from University of Waterloo. He worked as a research associate by Therapure Biopharma and as a bioprocess technologist by Sanofi Pasteur. He did his PhD in chemical engineering from Massachusetts, MIT, and postdoctoral research at Harvard University. He joined University of British Columbia in 2014. With this great background in academics and research, he also has several achievements to his credit. Some of those achievements are: in 2015, Wall Scholars Award from the Peter Wall Institute for Advanced Interdisciplinary Studies. In 2015, he received Teaching Excellence Award from Department of Chemical and Biological Engineering from University of British Columbia. In 2015, the Medicine Maker's Top 100 Most Influential People in Drug Development and Manufacturing. In 2014, from the same University of British Columbia, Green College Leading Edge Scholar. In 2012, ACS Chemical Abstract Service SciFinder Future Leaders of Chemistry Award. In 2012, winner of the Biochemical Society's International Science Communication Competition. In 2012, American Chemical Society Kaufman Foundation Entrepreneurship Prize. In 2011, MIT Legatum Fellowship for Entrepreneurship in Developing World. In 2010, Edward Merrill Prize for Outstanding Instructorship from the MIT. Sir, I'm delighted to invite and welcome to this international webinar on Frontiers in Science and Technology. Thank you. Over to Prashanthi.
Thank you, Jyoti ma'am, for giving very good introduction about today's guest of honor and valuable speaker. Now I welcome Professor Vikramaditya, sir. Sir.
Thank you so much, and thank you for the kind words. It's a special honor to present at a university that's named after Mahatma Gandhi, especially given what's happening in the world around us. His lessons are even more important, and they resonate even more today than ever before. So I hope you can see my screen,
yes, sir.
And can you just confirm if you're able to see my screen? I just see some yellow lines coming in, so I'll stop using the mouse. I'll just use my my keyboard. Okay, great. So
sir, I request you share your start video, sir. Your photo. Pic.
Can you see? Yeah, that's fine. That's fine. You don't. Okay, we can. I'll have to get my lights going here, but
okay,
hopefully.
Sir. Yes, sir.
Okay,
now it's
good.
Fine.
All right. Wonderful.
Thank you.
Okay. So good morning once again. So I am a chemical engineer by training, and my research group focuses on applying that engineering skill set and to design biological systems to solve some really important problems in manufacturing, in healthcare, and even looking at things such as infectious disease. I generally like to start my presentation by presenting an image that captivates people, or it really jars people. And I will get to why synthetic biology, it is not a panacea for all of the ills on our planet, but it is providing a very compelling solution to most of the problems that we face. And so the thing that I would like to start off with is this image of the polar bear in the very close to the North Pole. And this image, more than anything, is very succinctly capturing what our activities as society is doing to the planet. Climate change is a very real problem, and it is coming at a significant price to the environment. But despite all of this industrial activity, despite all of these profound changes to the planet, economically, we always seem to be very unhappy. And this is especially true now in a COVID year where we have seen that economic contraction. But generally speaking, despite all of this activity, we are unable to meet our economic aspirations. Now, this is just an image of Vancouver. It's a very beautiful part of the world where I am based. And the average monthly rent for just a one-bedroom apartment here is over $2,000. And that's just telling you how expensive the cost of living is. So we have to balance, as engineers and scientists, we have to balance these two polarizing powers. One is this rampant industrialization and the damage that it has caused on the environment. And the second is that the economy always seems to be working against us. And as engineers, this is that great balancing act, the triple bottom line as we address it. So our activities have to ensure that we continue to deliver economic growth. We have to ensure that this is occurring without destroying the environment. So environmental stewardship is really important. And ultimately, we need to provide high-paying jobs. We need to provide a high standard of living to every citizen of this globe. And meeting all three of these demands is going to be really important. And synthetic biology just happens to be one of many interesting technologies that seems well poised to deliver this objective, as I have stated it. And in my research group, we are a synthetic biology research group, but we draw on insights and innovations from a range of disciplines. And this, as was spoken in the introduction, I am a man of many interests, and my interests span everything from genome sciences and technology to green chemistry to even developing biosensors. And the examples that I will show you will try to address three distinct realms of activities that we are trying to do, and trying to address that triple bottom line as I have stated it. And so I will now get into those three specific examples, starting with one which is what industrial biotechnology is known for, is to develop bioprocesses and to develop processes that have high productivity. And I am defining productivity as nothing but yield per unit time of the process. So if you were to, in order to demonstrate this concept, I'm going to start off with a very simple diagram. And in bioprocess engineering, we typically deal with these three metrics: yield, titers, and productivity. Titers refers to concentration. So in your bioprocess, when you're producing material, you want to always have as high a titer or as high a concentration as possible. Yield refers to the production, so the amount of product that you have produced, and that is as a ratio with respect to the amount of substrate that you have fed to the process. So on a gram per gram basis, which is how we define yield, you always want to have higher yield. So you're producing more of the product per unit mass of the substrate fed. And in chemistry, we often allude to this as atom economy, so high atom economy. And productivity is nothing but yield per unit time or titer per unit time. Any one of those two units is good to define that. Now, typically in classic bioprocess engineering, when I start to improve a process, I am typically trying to improve the productivity. I'm introducing steps such as an online or an inline product separation, and that typically tries to improve the productivity of the process. But yield and titer are fundamental to that catalyst that we use in the process, which is the microorganism producing the material. And metabolic engineering or synthetic biology aims to improve exactly that. So in the triangle that you see to the left, when we use classic metabolic engineering, we are training that microorganism to very efficiently convert the substrate to product and withstand higher concentrations of the product. So you are driving up titer and yield. But in classic bioprocess engineering, you're always trying to improve the throughput of that particular batch. Now, you see that these two triangles are distinct, and there are very few processes that are able to achieve improvements in all three of the metrics in the same process. So it is this disconnect between the world of metabolic engineering and bioprocess engineering that we sought to operate in. So can we use synthetic biology to develop molecular circuits that are allowing the microorganism that we are using to produce the product of interest to sense the extracellular environment and then respond accordingly and improve the productivity? And this is what we call metabolic control engineering. So it's a molecular control of the process. And the way we actually did this, I'm actually just going to turn off the arrows. Okay, now this is clearer. Okay, so the way we did this is to design these molecular entities. And so the example that that we are using is to use a very special type of an enzyme. So we used enzyme engineering to change the T7 RNA polymerase. So for those of you who are familiar with the world of natural product chemistry, this is the non-mevalonate pathway. It commences with two central carbon metabolites, pyruvate and glyceraldehyde 3-phosphate. And then it ultimately ends in the production. So in the black that you're seeing, DMA, PP, and IPP, they're the universal terpenoid precursors that are then ultimately entering the dedicated pathway of interest. So in our process, I am engineering a microorganism to produce lycopene. Now, lycopene is the classic red color of tomatoes that you see. Now, in order to improve the productivity of this process, we decided to make the microorganism sense its environment. Now, this process, because of the nature of some of the enzymes, you are forced to operate the process at a low temperature. That ensures that some of these enzymes in the pathway, specifically ISPD, ISPF, and DXS, they are remaining soluble and therefore are operating at a high activity inside your cells. Now, when we lower that temperature, that comes at a cost, because the microorganism is now in an environment that it is not physiologically conducive to it. So therefore, it shuts down a lot of the other metabolic processes. So what we decided to use is to essentially take that temperature shift and use it to our advantage. So the T7 RNA polymerase, which is driving the expression of this downstream heterologous metabolic pathway that produces lycopene, we introduced a very specific type of a polypeptide sequence, which is called an intine. An intine is a self-splicing element. So when you are producing the entire protein sequence is produced, as you see, as a contiguous sequence. And as the protein begins to fold, when it reaches its desired conformation, the intine splices itself out and produces the active protein. So this is an example of a cis or a self. So from one single sequence, it is splicing out the intine. And we created a range of variants, one of which was active at 18 degrees Celsius. So what this enzyme engineering helped us achieve was to produce a T7 RNA polymerase with an intine sequence or an intervenin sequence that does not cleave and does not activate the enzyme unless the temperature of the fermenter is lowered to 18 degrees Celsius. So we have essentially created a thermometer and a metabolic controller inside the microorganism. And when we introduce this into the microorganism, you're seeing the results are very stark. So there are different variations of the intine we created. The one that is most interesting that you have to see, that you're looking out for, is the green trend line. And what you see at 37 degrees Celsius, there is no activity from the green line. And then what we are seeing, so this is essentially showing the adsorbance. So at the top, you're seeing OD, which is optical density, which is essentially telling you cell mass. But the one that you're paying attention to are the lower two curves. So this and this plot over here. And what you see is that you're seeing a flat green line, which is indicating no activity at 37 degrees Celsius. The moment you lower the temperature to 18 degrees Celsius, the intine splices out, and then you see this fluorescence. And this is comparing very well to the wild type intine, which splices at all temperatures. So it is essentially not discriminating based on temperature. And when we introduced this temperature sensitive intine into the microorganism, the results were remarkable. We immediately saw a drastic improvement in the productivity. So this example is the first of its kind where we are able to get the microorganism to automatically detect what is going on in the bioprocess and then start its cellular processes. And being engineers, we have modeled this, we've modeled the kinetics. So what you see over here are just different kinetic parameters, and that is informing future iterations of the enzyme engineering to improve the productivity further. Now, as you're seeing the difference between the green and the typical bioprocess, so this is the best in class reported by University of California at Berkeley, and our process is much better than theirs, and it's in a very short period of time, it's already 15% better. So that's just telling you the importance of designing molecular switches that can sense the extracellular environment. We have since gone on and had a generation 2 of this molecular switch. So in the previous example, I talked about temperature. What we have since done is gone and made a switch that is able to sense and detect light, and based on photostimulation is able to turn on or off the circuit. And this is just a very complicated example of how the circuit actually works. But to distill this entire complexity down into one single statement, when we shine light at 660 nanometers, we are activating the production of the active T7 RNA polymerase, which then eventually leads to the production of lycopene. And what you're seeing now is precisely that. So when you are illuminating and you're adding some of the other inducers to trigger production, you're seeing a drastic improvement in production compared to systems that are not able to do this. And we have then gone on and optimized the system. So the optogenetic circuit, this is one of the very first demonstrations of an E. coli that can sense light and can use the light trigger to control its intracellular metabolism. And as you can see, so without belaboring the point, the circuit performs as we intend it to do, and we are able to even identify and play around with parameters such as when to trigger the light, how long you should trigger the light for, and whether you should use something like an LED or a laser or different source of illumination. So this example is essentially telling you how we are developing cells that can sense their environment. And the potential impact of this is quite significant, because now we are in a position to improve the productivity of biomanufacturing processes. And that is a huge win for the industry, because now by operating processes for a short period of time, you're able to actually produce much more product, which is a fundamental advance for the field, especially when it comes to industrial biosynthesis of some of these value-added terpenoids, many of which are important pharmaceutical and active pharmaceutical ingredients. So this is what we now possess, is a very interesting toolbox of molecular switches at our disposal. Now, speaking of lycopene, now this was the first story, improving the productivity. So when you have more productive processes, you have to run them for shorter, so you are reducing your carbon footprint. Now, is there something else that we can do? So the natural extension of this system, it was producing lycopene. Lycopene itself is a photoactive dye. And we thought to ourselves, can we actually extend this and use that lycopene in a different way. So lycopene is conventionally used as a nutraceutical. Can we find interesting applications for lycopene? Because now we have cells that are making a lot of lycopene. And we have been able to construct bacterial solar cells using some of these bacteria that are producing a lot of lycopene. And we have essentially initiated an entire field of biogenic solar cell or biogenic photovoltaic materials. So this entire field of energy biomaterials has now opened up for us, where we are now making a lot of very interesting advances in this field. So to give you just an example of that, so you take that previous image of the bacteria that is producing a lot of lycopene, and now lycopene being a very photoactive or a very light sensitive molecule, if you're able to shine light on this, lycopene can then essentially break down, produce electrons, and you could use these electrons to power an electrical circuit. So this forms the basis of a dye-sensitized solar cell. Now, we can use the bacteria as is, it's producing a lot of lycopene, but the challenge with bacteria is that it is not a very good conductor. So what we used is we used supramolecular chemistry to attach, so using tryptophan linkages, attach titania onto the surface of the bacteria and thereby create these extremely porous and highly conductive biogenic electrodes that we are using at the anode. And this is now able to power our bacterial solar cell. So what you're able to show, so just to show some confirmation of the XRD spectra, just to show that we are producing an entirely new class of biogenic materials. So the system is bacteria, you're adding tryptophan, you're adding titania, so you're using aqueous-based chemistries, very clean chemistries, to then produce these titanated bacteria, which we then essentially calcine to produce extremely porous electrodes. So this synthesis scheme that I've just defined is one of the cleanest ways to produce a photovoltaic material. And just to confirm that what type of electrodes we are seeing, so this is just telling you this is how the bacteria is forming these very interesting materials, and this is what the electrode looks like. So high surface area, extremely porous, extremely lightweight, can be deposited onto any surface and any geometry of your liking, and it produces a remarkably versatile solar cell. And so now what you're seeing is now in amongst biogenic materials, Gratzel was the first one who described such dye-sensitized solar cells, and a research group at MIT has since developed a version of the Gratzel cell that is achieving a current density of about 0.5. This biogenic dye-sensitized solar cell that we have constructed uses aqueous-based, extremely clean chemistries, is a fraction of the time to build, and we are achieving a higher current density. So we are at a 0.686 compared to 0.5, which is what the MIT group was reporting. And what you're seeing over here is when we turn light on, you're seeing the current or the voltage, which is then driving the current. And then when you turn the light off, you're seeing this on-off switching behavior, which is telling you that the material is functioning the way that you want it to. And the Mercian Gratzel cell, which was from Berkeley, that has one of the highest open circuit voltages of 0.5, but it has a lower current density. So we are in that range. So we are amongst the best in class for these materials. And interestingly, for this type of material, what you're seeing is that it functions very well under low illumination conditions. So while solar cells are expected to perform well under well illuminated conditions, it's usually when there are overcast skies, when it's dark, when the light is not as good, that conventional solar cells don't perform well. What we are seeing with the bacterial solar cells is that they perform better under low light conditions. And this opens up, this property of these solar cells to perform better under low light conditions is opening up some very interesting applications in sensing. So you can imagine if a miner needs to go deep into some of these caves and requires low powered electronics to sense and detect the environment, run some very important analysis in the field, you can use these types of solar cells to power those devices. And this is the range of applications that we are currently seeking to develop. So this is very much ongoing work in the group, where we are looking at some very interesting low-powered electronics. But this is where synthetic biology has now gone from taking bacteria to producing optoelectronic systems. So the third theme, so I talked to you about improving processes, reducing your carbon footprint, taking solar cells from bacteria and producing some of the cleanest and the most efficient solar cells. Now, can synthetic biology solve some other problems? And one of the problems that we are very aware of in India, especially with India being one of the hotspots for malaria in the world, is this plague of mosquitoes. So mosquitoes have killed more humans in the history of humankind than any other serial killer, even more than Genghis Khan. Okay, so the mosquitoes really put Genghis Khan to shame in terms of number of humans killed. And malaria is truly a global scourge, and you can extend that to other mosquito-borne diseases such as Zika, such as the West Nile, the dengue fever. You're looking at all sorts of diseases that are essentially propagated by mosquitoes. So you're looking at about 700 million infections per year and 1 million deaths. And most of these deaths are concentrated in the developing world, in sub-Saharan Africa, a lot in India, a lot in Southeast Asia. So if we were able to halt the spread of these diseases in its tracks, that would represent a monumental victory for us. And one of the cheapest and the most efficient ways to stop mosquito-borne diseases is to use repellents. So as long as the mosquitoes don't bite you, they are not able to infect you. And typically repellents are discovered using this image that I'm showing to you before. So this is called an olfactometer, and an individual essentially places his arm inside this device. It looks like a very barbaric torture chamber, but this is precisely how repellents are discovered. And what happens is a drop of the repellent is added onto the individual's skin, and then he's praying to his god that let's hope that the repellent works, otherwise this entire swarm of mosquitoes is going to land on his hand and bite him. So this is how repellents are discovered. Now it sounds very, very cruel and barbaric, but the other and more relevant problem for the industry is that the method for discovering repellents, as you see on the screen, is error prone. There are a lot of false positives, a lot of false negatives, and you really cannot rely on these things. So testing, retesting, validation, that takes a very long period of time, and ultimately you don't get a lot of products making it through this development cycle. So is there a better way to identify repellents? And what we said is we can use that sense of smell of the mosquito, and if we can use or encode that mosquito olfactory system into a microbial host. So you take all the olfactory cascades as they are functioning in the neurons of the mosquito, you encode it into a simpler host, you can essentially develop a high throughput screen. And once you're in the possession of a high throughput screen, it opens up industrialized drug discovery. So you can screen millions and millions of compounds in a very, very short period of time, thereby improving the outcomes of repellent discovery and leading to more candidates translating to the market. This could be a huge advance for the field. It's a potential game changer for the repellents industry. And so the way we did it is we took that olfactory system of mosquitoes. And so what you're seeing to the left is the way the conventional mosquito system works. And then what you're seeing to the right is a simplified version that we used in our studies. But essentially, the way the mosquito olfactory system works is that the odorant first binds to a soluble protein called the odor binding protein, or OBP. The OBP essentially diffuses from the sheath all the way down. So this is a permeable cuticle. It diffuses through that space, and then it interacts with a complex of seven transmembrane proteins. One of them is your olfactory receptor, and there are about 79 in Anopheles gambiae, which causes malaria. And central to all of those 79, so all 79 of the olfactory receptors require an olfactory receptor co-receptor. or ORCO. And what happens is once OBP binds to this complex, it activates the complex, thereby converting it into a cation channel, and cations are then able to diffuse through that membrane. They're transported into the neurons. That thereby leads to membrane hyperpolarization, and then it triggers downstream olfactory systems that then allow the mosquito to understand what the odor is and then go and bite the host. So we took that system, but there are studies that have shown that the ORCO receptor by itself can also function as a cation channel. So the way, or a ligand gated channel as it is typically called. So the way we actually developed this system is we took this simplified system and introduced it into the yeast Pichia pastoris. And in the Pichia pastoris system, we then utilized that influx of calcium ions and we combined it with a calcium sensing fluorescent dye. So the Pichia pastoris is imbued with this calcium sensing fluorescent dye. You are expressing the ORCO protein on the surface of Pichia. When the odorant is able to go through the cell membrane and then it's able to bind to the ORCO on the cell membrane. It is then able to shuttle calcium ions from the growth media into the cell, and then it lights the cells up. So this is what that assay looks like. And what that ends up doing is depending. So now VUAA1 is a known molecule. This is one of the most potent repellents ever discovered. The only challenge with this repellent is that it cannot be volatilized very easily, so hence it was never commercialized. But we took this VUAA1, which was a known repellent, and when we expose it to this Pichia pastoris, so this is essentially a mosquito. We have converted the mosquito's olfactory system into a biosensor. So this is an example of a whole cell biosensor. And as we titrate the VUAA1, you're able to see this type of a response, and this is essentially the fluorescence. And this type of response is conventionally only measured using electrophysiology. So you need to use extremely complicated electrodes. You need to make sure that it is patch clamped properly with the neuron. So they're very, very challenging experiments. In our very simple experiment, we're taking the genetically engineered Pichia, we're putting it in a calcium-rich media, we are adding the repellent, and the Pichia lights up with fluorescence. And depending on the concentration, you're seeing this type of a response. And if I were to increase the extracellular concentration of calcium from 1 millimolar to something higher, you're seeing that the circuit starts to respond accordingly. And these type of responses are classic in olfactory systems. So anybody who's studying the sense of smell will look at these curves. They're called TRP curves. And this is how we are generating it. So we have recreated the mosquito sense of smell inside the yeast, and we are able to do all sorts of EC50 measurements to find out the kinetics and to find out what the EC50 values are. And ultimately, what we now have is a very, very good biosensor for mosquito behavior modifying compounds. When we expose it to citronellol, we are seeing citronellol is one of the most celebrated repellents on the market. We are seeing a complete suppression of the activity. And then when we finally add some VUA later, we're able to revive that activity. So we're seeing that interesting receptor dynamics at play. OCT1N3-ol is a molecule in human sweat that is a known attractant of mosquitoes. It is not shown to ever bind to ORCO, and we are able to confirm those results. So whatever results people have painstakingly studied in mosquitoes over several decades, we have been able to replicate in our system in a matter of weeks. And we now possess one of the most powerful biosensors to discover repellents. And this opens up some very tantalizing visions of using high throughput repellent discovery to introduce more products into the market. We just don't have to deal with Odomus. We can have a range of other different products. So I need about 2 minutes. I realize that I'm very close to time. This is just leading to some of the work in environmental remediation that my lab is doing. So in addition to my job as an academic, I also am the chief technology officer of a startup company that is commercializing some of these synthetic biology solutions that we are developing. And in this case, we are attempting to solve one of the largest environmental disasters that could happen in the next 30 years. And this is the problem of the oil sands tailings in the province of Alberta in Canada. So the oil sands have one of the largest deposits of oil. It is essentially a frozen mixture of oil, sand, and ice. And people are developing all sorts of treatment techniques to recover the oil. An unfortunate byproduct of that process is that you produce a lot of contaminated water, and you need to treat that water. Otherwise, you're going to have an environmental disaster. So I need about 1 minute, and I'll just summarize the story very quickly. So there are about 1 trillion liters of this highly contaminated water that needs to be treated. It is very expensive to treat, and given the large volumes, you cannot use something like advanced oxidation or UV. So we are engineering microorganisms to actually be released in these ponds and passively treat the water at a very low cost. We have performed a range of experiments. So current concentrations are 100 milligrams per liter. We require the 100 milligrams per liter to go down to about 1 milligrams per liter. So about 100x reduction in concentration of naphthenic acid. And you can see these naphthenic acids, they are an extremely complicated chemical class. What we have done is we have gone to these environments, we have discovered new strains, and now we are genetically engineering those strains to improve the production, improve the degradation of naphthenic acid. So I will quickly rush through these slides because there's a lot of systems biology detail, but we have uncovered the metabolic networks. And now, as I speak, there is a large-scale field trial that that my startup company is running, and we are hopeful of developing a microbial solution to treat this water in the near term. This is a huge problem for the country of Canada, and if this technology works, we could be potentially releasing such microorganisms to treat other industrial wastewater sites, including, for example, the Ganga River. So with that, I will stop. All thanks for all these brilliant ideas, and all the work goes to my very talented group of research students. And I will stop for now and field a few questions if there are any. Thank you so much for your attention.
Thank you very much, sir, for your wonderful lecture about the challenges using synthetic biology. Really, with your lecture, you have taken a world of knowledge. Thank you so much, sir. I request participants, 1 or 2 questions are allowed within a minute. If anyone, please unmute yourself. 1 or 2 questions from participants. Please unmute yourself. 1 or 2 questions are allowed.
Good morning, sir.
Good morning.
Sir, why you only choose the lycopenes only of your choice? Why don't the other xanthophylls in the plants? There are so many pigments. So why you choose only lycopenes?
So the answer to your question is, I just showed you an example of lycopene. We work with a range of different molecules. In fact, we work on a range of different terpenoids. So you have carotene, you have all sorts of different carotenoids. So and many of them are actually very interesting molecules. So we do look at those molecules as well. I just didn't show some of those results. And I think in that, if I were to guess the number, there's quite a few to keep a track of. We're working on about 10 to 12 different molecules in that terpenoid class. And the choice of those molecules, it just depends on what the industrial application is. If it's and we typically, something I didn't mention is we collaborate extensively with industry. So every project I presented, we work with an industrial partner to commercialize it. So either we start companies or we work with companies. So the choice of the molecule usually comes from the company. There is a good business case that's backing the molecule. But you raise a very, very good point. In the nutraceutical class, lycopene is not as used as some of the other xanthophylls, and so that is something that you've raised a very good point.
Thank you, sir. If the same Jivo plant lycopene and the wild lycopene has any same effects or any other impact.
So they are identical. So the lycopene that we produce is identical to the tomato plant. So it's a small molecule. We are able to do NMR and confirm that it is identical to the plant system.
Okay. Thank you so much, sir. Thank you.
Thank you very much. Thank you very much, sir, for your wonderful lecture on biogenic solar cells and bacterial solar cells. Sir, actually, which is the best material, sir, among all what you have done so far?
So far, I'll tell you. So solar cells is a broad field. So the conventional cells, they use silicon, and silicon cells, they work quite well. Now, silicon has some manufacturing challenges. So especially when you're trying to mass produce these things, you are able to produce it quite cheaply, but there is an environmental cost associated with silicon. In the world of biogenic solar cells, people have used dye. So they would extract the dye from plants. They will introduce this into metal organic type systems. So titania is generally the type metal oxide. Sorry, so titania is usually the one that they use. So there's a lot of biogenic or biological systems. Ours is a truly biogenic. It's based off of a bacteria. So the generation 1 solar cell that I showed you, we have since constructed a generation 2, where we have genetically engineered the bacteria further to actually grow iron. So the pili that cover the bacteria, the appendages of the bacteria, we have essentially genetically engineered that to incorporate iron. So we make the bacteria even more conductive. So what you see is that you see this porous network with a lot of micro wires extending in all directions. And we have seen almost 50% improvement in the performance of those cells. So all the work that I've presented has been published in journals. This one that I just mentioned to you is something that we are in the process of preparing for a publication. But amongst the biogenic solar cells, that's something that we have looked at. There are other groups that have simply focused on the dye. So they are producing all sorts of different dyes or extracting them and introducing them into a conventional dye sensitized solar cell.
Thank you very much, sir. And we highly impressed with your startup companies. Sir, thank you once again for giving a consent to deliver your talk during this pandemic. Thank you very much, sir.
Thank you. Thank you for having me. It was a pleasure presenting, and I wish the university all the best for all your future endeavors.
Thank you, sir. We will welcome you all in our future webinars.
Indeed. Thank you so much.
Thank you so much, sir.
All right. Bye.
Bye, sir. Thank you. Now, I request today's guest of honor and valuable speaker to our event, Professor Sangilhan, sir. Actually, due to unavoidable circumstances, the talk will be presented as a recording. For that, I invite Mrs. Mahati from Department of Chemistry and Pharmaceutical Sciences. Please introduce, sir. Over to Mahati. Mahati? Mahati is there?
Yes, ma'am.
Yeah, Mahati, please introduce today's guest of honor, Professor Sangilhan, sir. Just you raise your voice, please.
Good afternoon, one and all.
Yeah. You increase your volume, please. Volume is very less.
Okay, ma'am. Am I audible, ma'am?
No. No, please. Mahati?
Ma'am, am I audible, ma'am?
Yeah, little bit. Just increase your volume. Yeah, you proceed.
Yeah, good afternoon, one and all. I am Mahati, Department of Chemistry and Pharmaceutical Sciences, Mahatma Gandhi University, Nalgonda. It's my honor for me to introduce today's speaker, Professor Sangilhan, for today's presentation. Professor Sangilhan is currently Associate Professor of Engineering, Changwon National University, South Korea, since 2014. And Professor Sangilhan earned his Bachelor of Chemical Engineering at Pusan National University in 2003 and his PhD in Chemical Engineering from the Virginia Polytechnic Institute and State University in 2010, and working with Professor Stephen Martin for the study on organic solutes transport in liquid crystalline membranes. After completing his PhD, he continued his postdoctoral research in School of Chemical and Biomolecular Engineering at Georgia Institute of Technology with professors Carson Meredith, David Scholl, Krista Walson, Shankar Nair, Christopher Johns, William Corus for carbon dioxide capture project using metal organic frameworks. And he moved to Georgia Rutledge Lab in Chemical Engineering at Massachusetts Institute of Technology in 2012 and focused in fabrication of thermoregulated membranes using electrospinning techniques for cigarette development funded by Philip Morris International. So Professor Sangil Han offered his services as Director of Academic Affairs in Korean Institute of Chemical Engineers in 2018 and presently he is working as Director of General Affairs in Busan Korean Institute of Chemical Engineers and as Director of International Affairs in Division of Separation Technology of Korean Institute of Chemical Engineers. And it's my privilege to invite today's speaker, Professor Sangil Han, for today's session. I welcome you, sir, and I thank my organization committee for giving me this opportunity to speak few words. Over to Prashanthi.
Thank you, Mahati, for giving very good introduction about today's guest of honor. But due to sudden unavoidable circumstances, sir has sent his record. Now I request technical team to share Professor Sangil Han's talk.
Hello, everyone. Thank you for inviting me for this opportunity for the conference. In this presentation, I would like to present the recent research achievement in our group. So in our group, we are synthesizing the metal organic frameworks, which is called the MOF. So by synthesizing and developing the MOF, we are planning for the CO2 capture. Also, currently we are working for the VOCs removal. This is my research conducted during my postdoc at Georgia Tech Chemical Engineering Department. So I want to talk a little bit my previous research. So during my postdoc, I developed the high throughput screening system that is which is developed to screen or to select the best candidate move for short capture. So as you can see, we synthesized different several moves, and by using this high throughput screening system, which measures the CO2 adsorption capacity and the nitrogen adsorption capacity, and resulting in the CO2 capacity and the selectivity of the CO2 over nitrogen. By using this system, we selected a few the best candidates for developing the mixed matrix membranes. In this mixed matrix membranes, this is the cross-sectional image of the membrane. In this membrane, the inorganic morph particles are dispersed through the polymer networks. So when the CO2 or nitrogen, the mixtures are transferred through the membrane, only the CO2 is, we expect CO2 is passing through faster than the nitrogen because of the size difference of the molecules. The MOF is porous materials. So porous materials usually have the high surface area and the high pore volume. But in the metal organic frameworks, the pore internal structure is very well organized. So that's similar to the zeolite. And zeolite is also porous material, has the high surface area. However, the activated carbon, even it has a high surface area, but there is the pore is amorphous. So that means the crystalline structure is there is no crystalline structure, and the pore is randomly organized. So according to the IUPAC, the class, the porous materials are characterized in 3 different class: microporous, and mesoporous, and the macroporous, depending on the pore size. In the MOF, zeolite, and activated carbon, those are classified as microporous material. So I would like to talk about little bit about the metal organic frameworks. The metal organic frameworks is composed of metal and organic linkers by coordination bonding. So if you see this picture, the blue is a metal, and this metal is connected by the organic linkers. So those for each MOF, the internal structure are different, and this internal structure is continuous through the material. This yellow ball means that there is a porous pore structure. So depending on the different combination of metal and the organic linker, it has different surface area and different pore size. So generally, it has a larger surface area than zeolite and well-defined pore size. Also, the good advantage is there is no dead volume. For example, for the zeolite or the activated carbon, there is a dead volume, which that means the pore is not open. Pore is not fully open structures through the material. So even there is a pores in the zeolite and the activated carbon, the other side is sometimes the other side is blocked. Due to the disadvantages of MOF, it has used a lot of different applications. So, separation, hydrogen storage, and catalysis and sensors, the electronic materials. So this shows how to design the MOF, how to design the MOF structures. If you see that this is green is a metal, and this one zinc oxide is organic linkers. Green and zinc oxide. Those are those form the metal centers, and this yellow is organic linkers. When these 1 and 2 combine together with different linkers, when the linker length is increased, the pore size is also increased. So this is how we can tune the surface area and pore volume and pore size of the metal organic framework. There are more than 4,000 the known crystalline structures. So in this below picture shows the different kinds of the organic linkers. But even that the structure is known, the performance such as the adsorption capacity and the selectivity is not known yet. So in our group, we discover the performance of the known MOF, or we synthesizing the new MOFs. Okay. So for our research, for my postdoc research, the first step is based on the DFT computer simulation. This is the first screening process. By using this computer simulation, we selected 10 or 20 MOFs. And then based on the selected mobs, we synthesize the selected mobs, which is suggested by the computer simulation based on the DFT. On the DFT simulation, we consider two different poles. One is pole emitting diameter, that is the smallest poles. The other one is largest cavity diameter, that is the largest pole size. So pole emitting diameter is in the x-axis, large cavity diameter shows the y-axis. So in the for the selectivity, in terms of selectivity, pole emitting diameter is much important than the large cavity diameter. Because if you see the carbon dioxide kinetic diameter is 3.3 angstrom and nitrogen is 3.5 angstrom. But this carbon, the morpho pole size is PLD is between these two. Only CO2 is passing through the morph pole, but the nitrogen cannot passing through, cannot adsorb on the internal poles of the morphs because of the larger pole size than the larger molecular size than morph poles. That's why we selected the pole emitting diameter. Based on the pole emitting diameter, we selected the morphs. The pole size is between 2.3 and up to 3.6. So if we selected the already known morphs between this pole size, this is expected to show high selectivity, CO2 over nitrogen. So this slide shows the developed high throughput sorption measurement system. It composed of three different parts. One is lower side, lower chamber. The other one is upper chamber. In the lower chamber, this is the lower chambers. In this lower chamber, samples are loaded for different sample chambers. And this upper chamber is can be disassembled because after loading the samples, it should be tightly sealed by combining this upper chamber and the lower chamber together. And the sample chamber, the pressure transduce array is connected separately. If you see this, the below picture, all the the different sample chambers are connected with the different pressure transduce array. So when to for the measurements, CO2 gas is injected at the same time, while nitrogen gas injected at the same time. During the adsorption, this amount when this gas is adsorbed on the morph, the pressure of this air is decreasing. So by measuring the pressure changes, we can calculate how many CO2 or nitrogen molecules are adsorbed on the morph at the same time. This is the result by using the high throughput detection system. Those are the samples. For A samples, we can get this result at just with a single experiment. So based on this absorption capacity results, we got the absorption. This is the we selected these green mobs, but the red mobs were not selected. So we selected the green mobs based on the selectivity. sorption selectivity and diffusion selectivity. When this selectivity shows high values, we selected them mobs for the candidate for developing the CO2 nitrogen separation membranes. Also, we performed the SOx and NOx, the stability experiments. Because for the CO2 gas capture from the flue gas post-combustion the process, there is humid air and acid gas such as SOx and NOx. And it is known the MOF is some MOF is not stable when exposed to humid air and acid gases. So we wanted to prove our selected MOF is strong enough against the humid air and acid gases. So finally, the best MOFs were selected to produce the mixed matrix membrane, which is a hybrid polymer membrane where the inorganic MOF particles were dispersed like this. If you see this, the left image, when particle size is too large, there is a gap or defect between the particle and the polymers. This is polymer. When there is a gap or defect, the membrane performance is decreasing, significantly decreasing. So we have to avoid the defect or the gap between particles and the polymers. So by reducing the particle size is less than 1 micrometer. So when the particle size is reduced significantly, like you see this picture, the MOFs particles are well dispersed and there is no defects between polymer and the inorganic materials. So we also applied this cryogrinding method. On this cryogrinding method, liquid nitrogen is surrounding the MOF particle, which is contained in the reactor. So by using the liquid nitrogen MOF particle, the temperature was significantly decreased, and then large particles were mechanically broken. Like you see in this SM image, the before cryogrinding particle size was 400 micrometer. But after cryogrinding, about less than 30 seconds, less than 1 minute, the particle size was reduced a lot, 1 micrometer. Also, we checked the XRD data. As you can see, this simulated reference XRD picture, XRD pattern is all maintained even after cryogrinding. So there is no difference in the before and after cryogrinding. That means even after the particles are ground, the internal structure, crystalline structure is still there. But if there is no liquid nitrogen, just the grinding, all XRD peaks will disappear. So this was successfully done, and the ground MOF were mixed with Ultem polymers. Mixed with the Ultem polymers to produce mixed matrix membrane. So on this SM image, you see this MOF particles are dispersed, white MOF particles are dispersed. And this is the below picture shows that we made with a different MOF, COPCN and CONIC. The one shows the blue color, the other one shows the red color. Until now, I was presenting my postdoc research, and from this slide, I'll talk about my current research in our group at National University in Korea. So we are developing the new modes for the shot capture. We developed UTS-16 zinc and UTS-16 cobalt. Now this is the background for the why we have to do the CO2 capture. As you know, CO2 is a greenhouse gas which cause the major global warming problems responsible for climate changes. So it should be captured before releasing to the air. So there are known methods, known mechanism for the CO2 capture. One is absorption by solid, the other one is absorption by liquid, and also there is a membrane separation. But in our group, we are focusing on this adsorption method by using solid adsorbents. So our target is for the post-combustion, the CO2 capture. The advantage of post-combustion CO2 capture compared to other pre-combustion and oxy-fuel, though it can be used for the current power generation system without any large modifications. By the way, the pre-combustion and oxy-fuel, the oxy-fuel CO2 capture system or the oxy-fuel process, it requires entirely new process. It requires entirely new systems. So post-conversion is just simple compared to the other methods. But to make it feasible, to make it more feasible application to the current capture system, the solid adsorbent performance should be improved. So that's why we are focusing to develop the materials which have high capacity, short capture capacity, also CO2 and nitrogen selectivity. We use the microwave synthesis system. In the microwave synthesis system, the organic linker and the metal was reacted to produce the porous crystalline adsorbent under the microwave irradiations. The advantage of using microwave system is the reaction time is reduced. That means it provides us the faster reaction. And the produced move particles are very uniform. So that means the particle size are very uniform, and also it has a high quality of the crystalline structures. So for we tried two different methods to develop the microwave synthesis method. Solver thermal method should be proven for applying the microwave synthesis method. So solver thermal synthesis method, citric acid and KOH and ethanol cobalt acetate were mixed together, and then in the autoclave solver thermal reactor under the 120 Celsius and for more than 48 hours, it was reacted. So it requires quite a long reaction time. But in the microwave system, we are using the same solution, reaction solution, but under the microwave heating, it's just done less than 12 hours. In our research, the best performance showing material was synthesized in 4 hours. So this was published in the Chemical Engineering Journal recently. So also we replaced the cobalt metal with zinc on the previously developed UTH-16 MOF. So the cobalt precursor was replaced by the zinc acetate dihydrate. So under the similar condition, we produced the UTH-16 zinc. This material is the new material. But when you compare the crystalline XRD patterns, it shows the similar XRD patterns. That means only the metal was replaced on the same crystalline structure. So to measure the adsorption performance, short adsorption performance, we use the two different systems by using based on the volumetric method using Bell-Solve Mini, and for also using the gravimetric system by using the TGA. So with the volumetric method, we can produce the adsorption isosum. By using the gravimetric system, we can provide the adsorption kinetics. Kinetics means the adsorbed amount is a function of time. So as you can see, the XRD image, all XRD's image, XRD pattern are identical. Even the 1 hour reaction, 2 hour, 4 hour, 6 hour, it showed all the similar XRD patterns. That means even with the 1 hour small reaction, crystalline structure was formed. But in the next slide, depending on the reaction time, it shows the different the adsorption capacity and the adsorption selectivity were observed. Also, for UTH-16 zinc, we selected 4-hour material because it showed the best performance. For the stability test, after SOx and NOx and humid air exposure, as you can see, all XRD pattern is still there. That means it's stable under that harsh condition. Those images shows the those are SM images showing the particles size MOF UTH-16 zinc particles with a different time. So when as the reaction time increased, particle size were decreased. Also for UTH-16 cobalt, the similar pattern, similar behavior was observed. As the reaction time increased, 1 hour, 2 hour, 4 hour, 8 hours, the particle size were decreased. And this first image shows the solvasomal kinesis made by the solvasomal kinesis reaction. For the same material, but depending on the kinesis method, it shows the different particle shapes. So as you can see, the materials made by the microwave method, it shows the more uniform structures and the uniform shape. Here shows the surface area results. It was measured by using the BT instruments. First table shows the UTH-16 cobalt. Second table shows the UTH-16 zinc. When the reaction time increasing, the BT surface area was also increased for both materials. But it shows the maximum surface area and the pore volumes when the reaction time was 4 hours. After 4 hours and 5 hours, 6 hours, the surface area and pore volume were decreased again. So that means the 4-hour reaction time was optimal. So this shows the the stack of the UTH-16, the MOF. So UTH-16 cobalt. So which image, this image was obtained from the journal. That is the original journal which they introduced the UTH-16 cobalt. But in our research, we synthesized and in the first UTH-16 MOF was synthesized by solver-thermal reaction. But we developed the UTH-16 MOF by using the microwave. Also, we produced UTH-16 zinc for the first time from our group. So this shows the capacity, CO2 and N2 capacity. As the reaction time increased, the CO2 adsorption capacity was increased. At the green, 4 hours shows the maximum. And the nitrogen, the advantage is, the good thing is, when the reaction time is increasing, nitrogen adsorption was increased, but in the 4 hour, not increased continuously, but in the 4 hours, it decreased again. Even at the 4 hours, the surface area and pore volume are still increasing. So which gives us the better adsorption nitrogen, CO2 and nitrogen adsorption selectivity, as well as high CO2 capacity. In terms of the stability study, for the UTH-16 cobalt, after the NOx exposure and SOx exposure, the CO2 adsorption capacity was decreased. But for UTH-16 zinc, the decrease was much less compared to the UTH. So cobalt material, after SOx, the capacity decrease was 40%. But the zinc material is just less than 10%. So our new developed UTH-16 zinc material shows the similar very high CO2 adsorption capacity, as well as the improved the stability to humid air and acid gases. That's very important. So this is our conclusion. UTH-16 zinc, when made with zinc metal, this shows the better performance in terms of the stability. This shows less capacity decrease than cobalt UTH-16 cobalt material, but also it still maintained the exceptionally high CO2 adsorption capacity, like UTH-16 cobalt material. Also, we evaluated the cost analysis for production of the UTH-16 zinc material. So for this blue, each UTH-16 zinc, the y-axis is a CO2 capture capacity per unit production cost. That means in the high value is better than the others. We compared with other already published, already reported MOF materials. This is better than the other materials. Also for the selectivity, CO2 and nitrogen adsorption selectivity for production cost. So this is exceptionally high than other already reported material. So as a result, this developed UTH-16 zinc material provides high CO2 capacity and the adsorption selectivity. Also, it can be synthesized with very low materials cost. So as a conclusion, the bimetal exchange from cobalt to zinc, we produced utilizing zinc materials, zinc metal organic frameworks. And the CO2 adsorption capacity, it still shows the among the best performance among the other reported modes. Also, the advantage of this material is it can be synthesized with a very low material cost. Also, reaction time was significantly reduced from more than 24 hours to 4 hours. Thank you very much for this for listening this presentation. If you have any questions, please send me emails. My email address is right here. sangil.hahn@gmail.com or shan@changwon.ac.kr. Thank you again for this listening.
Thank you, sir. During this pandemic, sir has given his great concern and commitment by delivering the talk on recording. So I, on behalf of this international webinar, I convey my sincere thanks to Professor Sangil sir for giving very good lecture on metal organic frameworks. So now this is the time to move for the oral presentations. So I request, I will announce a few names of participants. First speaker will be Dr. Ravi sir, Research Professor, Changwon National University. Next, Dr. Sangeeta Kulkarni ma'am. Next, Dr. Pooja ma'am, Dr. Lakshmi ma'am, Dr. Ramarao Garu, Mr. Hari Prasad, Mrs. Abhinya from Coimbatore, and Mr. Ambati Chandrasekhar from US. So I request all of you be ready with your presentations, and the presentations will be around 5 minutes, 5 to 6 minutes. For the first session, 3 to 4 members, I request Dr. Annapurna ma'am and Dr. Ramesh Domala sir to chair the session. And for this, please welcome ma'am for this session. Now, I request Dr. Ravi sir from South Korea, Research Professor, Changwon National University. Please, sir.
Yeah, I'm here, Prashanthi.
Yeah, ma'am. And Ramesh sir, please welcome for this session. Dr. Ravi sir.
Madam, good afternoon.
Okay, sir.
Yeah.
Good afternoon, Prashanthi.
Dr. Ravi sir. Dr. Ravi sir, Research Professor from Changwon National University, South Korea. Sir, may I know, sir?
Dr. Ravi.
Dr. Ravi, sir?
He's not there.
No, ma'am. Sir is available. So I think we will go for next. Dr. Sangeeta Kulkarni, ma'am.
Hello. This is Ravi from South Korea.
Sir. Yes, sir. Please welcome, sir. Please.
I am here now to present.
Yes, sir. Please share your screen, sir. You can start. Sir, I request you to conclude your talk by 15 minutes.
Okay.
Thank you, sir. Thank you.
From South Korea. Not him.
You open it. Hello.
Hello, sir. You are audible, sir.
Yes. Can you see my presentation lecture?
Yes, sir. Just it's started.
Yeah.
About to start, sir. Yeah, it's there, sir.
Yeah, this is Professor Ravi from UNIST and Changwon National University, working with mechatronics and mechanical engineering. So presently, I would like to present my ongoing research work is harvesting of energy through self-powered triboelectric nanogenerators. So here, what is the triboelectric nanogenerator that I wanted to present here? So presently, the triboelectric nanogenerators are very trendy research area because the harvesting of energy is very important to industry, domestic, and public purposes. So we are working on several years to develop a various nanomaterials for generating of the nano surfaces and also harvesting of energy through triboelectric charges. So what is the triboelectric charges? This is the self-induced power when it is interact with the opposite charge when compared to the lower energy to higher energy. So the harvesting of energy is mainly several types. So I divided my contents into introduction, what is tank and history, modes of tanks, application of tanks and problems to be discussed, what is present work and results and discussion, and what I have developed various nanomaterials here to generate the power. So if you compare with the introduction part, a nanogenerator is a type of technology that converts mechanical energy or thermal energy into electricity. So a nanogenerator has typical approaches which are mainly having different pico or pyroelectronic charges. So the history is mainly depends on the uh, Professor Wang, who is Georgia Institute of Technology. He is the inventor of the triboelectric generators. He is the pioneer and he developed various systems to generate the power. And he is the real researcher who published more than 2,000 publications in this area. So he is the person who is standing with me in the slide. He's the Professor Wang. And what is the triboelectric nanogenerator? How it works? So that converts the mechanical energy into electrical energy based on nanostructures, which is piezoelectric material or triboelectric materials having here. So you can see this image, the zinc oxide nanorods have been touched to the opposite surface. This opposite surface is generating the power when they occasionally touch and release this energy. This is called contact electrification process. This contact electrification process we have been developed in our laboratory. You can see the spring system. This is electric impulse spring that is having two different nanomaterials which are attached to the wire. That wire can be collected the surplus energy which is generating this nano surfaces. This is the triboelectric nanogenerator fundamental principle and which is mainly focusing towards that energy source. And how it works, actually the working of the triboelectric or piezoelectric nanogenerators, you can see this image, the mechanical sonic vibration of flexible nanowires when touches the opposite charge. These two charges are, one is electronegative charge, opposite one is the electropositive charge. When they contact each other, automatically the charges will be generated. These charges are attached to the copper wire. That copper wire can transmit this energy which is from the system. This system will work continuously when it is continuously touch and release the power and opposite the charges, which is really incredible to generate the power or harvesting of the energy through this system. And also there are 4 different modes of triboelectric nanogenerators in the literature and also have been developed, which are mainly tanks or 4 types, which are vertical contact separation mode. You can see the image A, and in plane contact sliding mode. And we can just slide the image and you can generate the power through sliding mode. And single electrode mode, we can just take the energy through single electrode mode, and freestanding triboelectric layer, which is generally passed through the air. So the freestanding is almost the electrical wires can be, or the electrodes can be touch each other occasionally, then charge will be generated, which is mainly used for the uh, working style, a triboelectric nanogenerator and electric trains, such as several things you can use in the domestic way. And the principle of this electrode concept is mainly contact separation mode. You can see the upper electrode is generating positive charge and lower electrode is generating negative charge. These two electrodes, which are mainly developed for PTFA and PVDF materials, and the electric positive charges we can generate through our laboratory. We can generate a nano surfaces which are mainly useful for the generation of the triboelectricity at the tip of the surfaces. These are very minute, and also you can observe through a SEM electrode or scanning electron microscope procedure. And you can see the power output from nano generator. You can observe this equation. This equation can generate the power V maximum, that is equal to plus or minus 3 by 4 into K by K plus. And you can see the following equation. These are the main materials we use in the inorganic size, that is zinc oxide, cadmium sulfide, gallium nitride, and also lead, zirconium, and titanium oxide along with the oxygen. This is PZT material, which are very useful for the generation of the power from 0 to 65 millivolts. And what are the problems to be discussed in the triboelectric nano generators? This is very important and the key features we have developed. So the fundamental mechanism already I explained, that is contact electrification and the quantitative understanding the surface charge density. If the surface charge density is very high, you can generate more power from the surface. So my role is we have to generate more density power and high surface area power nanomaterials that we are generating in our laboratory. And also measurement through these materials, and the choice of the materials are also very important, and the power management. So when you generate the power, how you manage the power, that is very important, and the durability and the stability. Even though you develop a material, if the material is not durable, this is not useful for the further materials and also further generation of the power. And the packaging also very important. Many haptic devices such as cell phones, there is a problem called packing system. The battery is not packed very well, automatically the cell phone will not be work for a long time. So the wastage of energies you can occur from this packing system. And also energy storage also very important. And continuously you can find hybridized cell for harvesting of types of energies, and hybridization with the traditional electromagnetic induction generators also very important. These are the problems we have to face now in the domestic purpose and industrial purpose. We are developing these kind of generators to solve these kind of issues. And the generation of power from various tanks, you can see here this tank introduction and tank importance is gradually increasing since 5 years. And you can also see vertical contact tank, which is generating almost 313 watts per meter square. And the freestanding mode, you can see 1.2, and the single electrode mode 0.5 watts per meter square, and the lateral sliding mode you can see up to 5.3 watts per meter square. And also we can see the domestic way sensors we can attach to the hands, and also heart, and also muscles to the human, and brain sensors, and also pacemakers we can use for the generation of the harvesting of energy or nano generators through these materials. And recently I published in Nano Energy electric impulse spring assisted triboelectric nano generator, which is fabricated from polyaniline emerald salt and woven carbon fiber. So this is a very important subject we developed recently and published in Nano Energy. You can see the stage 1 and stage 2 of the figure 1. We have developed this material using simple carbon fiber and oxidized by Piranha solution by harsh oxidation method, and we generated and deposited the polyaniline emerald salt on top of the carbon fiber. This carbon fiber is strongly attached by dip coating method or drop casting method. After that, we use this material as a electropositive electrode. That electrode can be touched with the PTFE or PVDF. That material can be, you can see here in the image, there are two. White layer is the PVDF, and green layer is showing, you can see that is electropositive layer. The pillars have been deposited on top of the PVDF, carbon woven carbon fiber surface. So these nano pillars, when touched to the surface of PVDF, we generated power in different combinations. We obtained almost 90 volts of energy from the contact separation mode. And you can see the results, results of output voltage before and after deposition of carbon woven fiber with polyaniline emerald in salt. After deposition, we generated 3 different oscillations of the speed. When the speed increases, the power also increased. We generated the power almost 90 volts and 120 volts, and we generated the current almost 3.5 microamperes. This is the useful current for the industrial and domestic purpose to use it. And also we tested this material for a long time, almost 10,000 cycles. We got continuous and stabilized material, and the current also obtained continuously with this material. And also the voltage also constant we observed at 5 hertz of the volts. It's almost generated 95 volts as I explained here. And the other material we developed that is called dual demonstrative BNO SPI tanks for a strong contact electrification for vertical contact separation approach through ions and electron charge transfer. In this there is a specific introduction we have developed. There are 2 different materials inserted in one polymer, that is electropositive material that is acting with continuously by electrons and ions. When the ions moving, the charge is coming output from the harvesting of the energy when touched with the PTFE material. So you can see the yellow layer is the PTFE material, and there are 3 layers you can find, or 3 films you can find, that is BNO SPI SO3H and BNO SPI SO3Li, and also protected layer of BNO SP3I, that is triethyl ammonium oxide. So this material also very useful for the generating of the power through triboelectric nanogenerators. And you can see the results, the observed harvesting of energy at different volts, like different hertz. You can sign 3 hertz, 4 hertz, 5 hertz, and 6 hertz. The maximum speed we stabilized at 6 hertz, and we generated the power up to 1.2 microamperes. This is the maximum output we generated from the BNO SPA SO3H, because the H plus ions are very labile and they can easily move from the surface of the material, and when it is touched to the PTFE surface, automatically it can be generate the power at maximum. So the power density also very useful through this material. We have developed this material is using the oxidation of the various organic materials. Those materials are polymerized and generate the flat material. That material is flexible and also useful for the generation of the power through harvesting of energy. And there is another material we have developed through harvesting of liquid stream energy. This is also unsteady peristaltic flow, induced flow time. So we also generated power through water. So when you take a water through a surface of the electrode, the generation of power is very high. So this electric power we generated through like hydroelectric powers. This is a miniature power generator we generated and we developed it from the laboratory. So this laboratory technology we have been published in the Nano Energy. This is the 3rd publication through Nano Energy. Impact is almost 16.6. Recently it is hiked from 15.5. And this is the schematic information we developed here. You can see the peristaltic pump that is attached to the electrode and water reservoir. When the pump is moving, the water reservoir sucks the water through the electrode. The electrode will receive the water through the electrode. The electrode can generate the power when water is touches to the electrode. So you can see the electrode is measured through the Kettler instrument, and this is the power recovery machine. And also you can see the graph. So with the time increases, the power speed of the water is flowing on the surface, the charges also increases. You can see in the black and the red graph, the increment of the power also increases through this electrode. This is also a developed material we have published in Nano Energy in the 2019. And the mechanism of this generation of the power is, there is the electric double layer which is generated in the power line. When it is touches, the water is decomposed into H plus and OH minus ions. So H plus ions, when they touches this electrode in the inside of the pipe, and it is generating the power through the copper wire. We connect the electricity and connect to the Kettler instrument to identifying the how much energy we are generating to this electrode. And this is the real instrumentation we developed in the laboratory. You can see the water and electrode is attaching to the instrument, and also this is the peristaltic pump that can, when it is rotation, automatically the water will be pumped into the electrode and it is generating the power through this electrode. And also we studied in various combinations of the speed of the electrode through the electric pump. And when the speed increases of the water circulation, the output current also increases. You can see up to 20 volts we have generated from 1.1 volts. So the speed of the water is passing through the electrode. The generation of the output voltage is also very high. This is the evidence we published in the Journal of Nano Energy. And also recently I developed another material that is called sand polished Kapton film and aluminum as a source of electron transfer triboelectric nanogenerator through vertical contact separation mode. This is also recently published in International Journal of Smart and Nanomaterials. And this is the concept of our material. And also we see aluminum electrode and sand polished material before and after. After sand polishing of the Kapton film that is commercially available, that is very helpful to generate the power. And the project is already ongoing, that is fabrication of sand polished sulfonated polyimide using P.PSS as a conducting polymer, which is generating through various surfaces like sulfonic acid H plus ion and sulfonic acid H plus ion with P.PSSA, that is PP and SP for robust triboelectric nanogenerators, which is going on in our laboratory. I am the principal investigator to generate of this kind of materials. So I am the key material developer. So you can see here synthesis of sulfonated polyamide surfaces, which are strongly reacted with the amides like NTD amide, that is diamide or dianhydride, which is reacted with the two amines and get to the linear polymer. This linear polymer again hydrolyzed to get the carboxylic acid or sulfonic acid surface. This sulfonic acid surface we again lithiated by lithium hydroxide. We generate the lithium ion on surface of the material. This lithium ion is the crucial to transport of the electrons and charges through electropositive charge when it is touched to the PTFE or PVDF, and it can be generated the high voltage or high current when it is contact separation mode process happening.
Sorry for inconvenience. Can you conclude, sir? Sorry
yes,
for
this is the last slide. I am going to conclude about the schematic representation. We generated a different kind of surfaces for generation of the power. You can see the semi-maze. There is the hills and valleys you can find on the surface. And this is the mechanism we developed. And thank you for your kind attention and also paying attention for my research. I am really thankful to sharing my robust technology of triboelectric nanogenerator through this international webinar. Thank you for all. If you have any questions, please ask me.
It was very nice presentation upon triboelectric nanogenerators. Hope it will have a successful application in future for the world. Thank you for your nice presentation.
Thank you very much.
Anybody have any questions can ask by unmuting yourself.
I think you can ask me. I am ready to explain. I am well.
Actually, Ravi sir, really.
The main, I was thinking about the application because your research has given materials
yes.
which can generate power.
Yes.
What's the real-time application? That was the thing.
Yeah, that's a very good question. Actually, the real-time application we are using in the mechanical engineering applications, like moving of robots using this turboelectric nanogenerator. So we attach this nanogenerator to the robotic instrument. So the robotic instrument take this energy and it can move towards the application. This is the mechanical application we are already doing. And also we are applying for the haptic devices, that is called cell phones. So the cell phone also we can use using this technology. We are working on it in the future.
Pretty much charging the cell phone, sir.
Of course, of course.
Okay, good. Nice, sir. Nice work.
Yeah.
Thank you, sir. I already met you at MSSA.
Yeah, I know. I remember you. I think.
Yeah. Thank you, sir. Thank you.
Thank you very much, madam. Yeah.
All the best.
Thank you for the nice presentation and also giving me a great opportunity. I thank very heartfully to convener Y.Prashanthi madam. In the future, I can meet you if I have a chance.
Yes, sir. Thank you, sir. Thank you so much.
Yes.
Now I request Dr. Sangeeta Kulkarni. May I know ma'am is available?
Yes, ma'am. Yes, ma'am. Am I audible?
Yes, ma'am, I request you to conclude your talk by 5 to 6 minutes.
Okay,
Please
welcome. Please allow me to share the screen.
Yes, you can, madam. You can.
No, it is telling not other parties.
Your screen is there.
Sir, I think Ravi Kumar sir has to close the screen, then it will. Sir.
Hello.
Yes, ma'am.
I think he is not.
Ravi sir? Yeah.
Sangeeta ma'am?
Yes, I am ready, madam. He is okay.
You can.
Yeah, you can share the screen, ma'am.
Yes, I am sharing, madam.
After Sangeeta ma'am, I request Sanjit, Mr. Sanjit, South Korea from Changwon National University, be ready with your presentation for 5 minutes.
Please go ahead with your presentation. Thank you. Your screen is visible, madam. Sangeeta ma'am.
Yes. Myself, Dr. Sangeeta Kulkarni, and I am assistant professor in Government Degree College, Dharwad, Karnataka. Today, I am going to present on the effect of amount of graphene on the barrier properties of epoxy graphene nanocomposites. As there is a lot of work which is associated with these epoxy graphene nanocomposites, and especially in the barrier properties, only so many studies have been covered. But my paper concerned with the increase in the amount, whatever it has been studied in the past. I have chosen 0.75 and 1.58% of graphene nanocomposites to introduce into the epoxy films using dispersion method. This dispersion method attributes to the excellent water barrier property, which significantly increases the toughness and dampness of epoxy system due to the reduction in free volume with respect to the pressed-end polymer. And these graphene nanocomposites have excellent electrical, thermal, and mechanical properties. The electron transport, high surface area, and mechanical properties of graphene nanocomposites have wide potential applications in food packaging as well as in anti-corrosive coating properties. And after dispersion method, the synthesized films were characterized by differential calorimetric techniques, and the barrier properties were investigated using EIS, that is electrical impedance spectroscopic method. And in the results and discussion section, there is I have measured first the water contact angle for different epoxy graphene nanocomposites. There is an increase in the contact angle with increase in the amount of graphene nanoparticles, which is mainly leading to the reduction of absorption of water within the films. The measured contact angles were for pressed-in polymer, it is 66.6. For 0.75% of the nanocomposite, it is observed 88.1. For 1.58%, it is observed is 102.3 degrees centigrade. In the next part, I have studied EIS, electrochemical impedance spectroscopic results, which were mainly used to study the barrier properties. This study mainly includes the exposure of nanocomposite films to 3.58% of aerated sodium chloride solution, which gives the impedance modulus as in the form of result. This shows an increase in the increase in the impedance modulus when compared to no loaded epoxy system, which indicates the better barrier property, which was tested for more than 1 day also. After 1 day also, it remained same. And this increase in impedance modulus was mainly due to the formation of local anodic area for longer exposure of nanocomposites, say up to 21 days I have tested, which was attributed to the absorption of water electrolyte, which in turn shift the impedance modulus to lower frequency. This is the plot which I can show for the impedance modulus against or versus the immersion time. And the lower modulus, impedance modulus after 1 day, which could account for the poor barrier properties, which was observed for neat or pristine polymer. And this slowly increased after up to 21 days for pristine polymer also, but not that increase when compared to the embedded or loaded epoxy systems. And the observed increase in the impedance modulus was 8 into 10 raised to 7 ohm per centimeter for 1.5 as a function of immersion, which shows better barrier properties among the two. The compact behavior of these was explained on the basis of no full penetration of the electrolyte in the given time interval. The electrolyte was sodium chloride solution, which I have used for the exposure purpose. And in the next part, I have carried out my studies for calculating the diffusion coefficient, water diffusion coefficient. This is a plot of the mass absorbed against the square root of time for epoxy graphene nanocomposites as well as pristine epoxy system. And the calculated diffusion coefficient were increased. The high increase for neat pristine epoxy system, whereas these diffusion coefficients were decreased for the nanocomposites, which are synthesized from 0.75 and 1.58% of graphene. These coefficients were comparatively decreased for 1.5 when compared to 0.75 nanocomposite. And these are the values. I have calculated two sets of values. One was from fixed equation. Another was a time variable dependent, time variable diffusion coefficient equation. Both were showing the same result. And comparatively, the time variable diffusion coefficients were have a good agreement with my EIS values. EIS results. And the conclusion was the addition of nanoparticles to epoxy system lead to slightly increase in the hydrophobic character of the nanocomposites. And collected EIS data shows that loaded coatings exhibit improved barrier properties, which was attributed to the diffusion mechanism of water transport through the epoxy nanocomposites. And this is my thank you.
Thank you, ma'am.
And thank you, Prashanthi madam, for giving an opportunity to present. Thank you. Thank you so much.
Thank you, ma'am.
Thank you so much.
Any questions, anybody? Madam, regarding the contact angle measurements which I've done, one was 66, one was 102. Which was the best one, you think?
Which was? What
one was 66, right? The contact angle measurement.
66 was for, ma'am, 66.6 was for a neat or pristine polymer, which is comparatively low and which was not at all loaded with graphene. As I have loaded 0.75 and 1.58% of graphene to epoxy system, there is an increase in the contact angle, which shows the increase in the hydrophobicity.
Yeah, good. Nice one. Thank you.
Thank you so much.
Next, Mr. Sanjit from South Korea, Changwon National University.
Yeah, I am online now.
Yeah, so you can conclude your talk by 5 to 6 minutes, Sanjit.
Yeah, ma'am, sure. So today I am going to talk about different synthesis methods of MOFs. So the topic of my presentation is the rapid synthesis method of metal organic framework by using a micro irradiation for CO2 capture. So I am currently pursuing PhD in Changwon National University from Department of Chemical Engineering. So we are very well known about global warming, which is caused by a CO2 absorption. So which is caused by a CO2. There are different ways to reduce CO2 from the atmosphere. That is, first one is by using a non-carbon energy sources, as well as carbon capture and storage. There are different techniques that are used for CO2 absorption. That is, first one is absorption by solid. So next one is absorption by liquid, and third one is a membrane separation. So these are the three techniques that is by using that techniques we can capture CO2. So this is the techniques, that is micro techniques that I used for my research work. So there are some benefits of this micro techniques. By using this technique, we can make reaction faster as compared to conventional synthesis method. And also the product obtained after this reaction is a very less byproduct. Also, we can get the compounds or products in pure form. Also, the solvent used for this synthesis is, not hazardous like H2O, ethanol, and acetone. Also, quantity used quantity amount required for this synthesis is very low. So, in microsynthesis, generally it based on two mechanism, that is specific mechanism, that is dipole rotation and ionic conduction. So, in this micro method, there is a necessary between there is interaction between material and the oscillating electric field of microwaves. So this is a general method that all researchers used for the synthesis of this MOF UTSA 16, that is called as solvothermal method. This is a solvothermal method all researchers used for the synthesis of this MOF. By this, generally this method takes 48 hours to synthesis these MOFs, but our lab developed new method, that is micro method. We reduced the time from 48 hours to 4 hours by using these techniques. And the product obtained, final product UTSA 16 cobalt, shows a better CO2 absorption performance as compared to the MOF obtained by a solvothermal method. So this research work reported in the journal, that is Chemical Engineering Journal, last year. So another MOF we developed by using same technique, that is micro method, and that is the MOF name is UTSA 16 zinc. We replaced metal precursor cobalt acetate by zinc acetate dihydrate. Ligand and other solvent and other reaction mixture, everything we kept same. And we used reaction condition also same. So we developed a new novel MOF, that is a UTSA 16 zinc. And also we used this material for CO2 absorption. So this is a crystal structure of UTSA 16 zinc that we made by micro method. This is a structure of UTSA 16 cobalt. In our lab or in our experiment, we are performing CO2 absorption. Generally, we used two methods, that is volumetric method and gravimetric method. In volumetric method, we used Belstorp Mini 2, and for gravimetric method, we used a TGA for the CO2 absorption. So the evaluation of CO2 capacity. So all these MOFs that previously reported synthesized by a solvothermal method. So it's showing a less CO2 absorption capacity, that is less than 4 millimole. So we developed a new MOF, that new method, that is micro method, and it's showing better CO2 performance as compared to the research solvothermal method made by other researchers, that is around 4.5 millimole per gram. So this is our good achievement, that we reduced the time from 48 hours to 4 hours, as well as CO2 absorption performance of our material is better as compared to previously reported UTSA 16 cobalt made by a solvothermal method. As well, if we compare our lab-made material with other MOF, this four are related to UTSA 16. But if we compare with other MOF, that is HCUST, UIO, MIL, and GPET, it also showing better CO2 absorption performance as compared to other MOF as well. If we see the SEM image, that is, this is a novel MOP UTSA-16 zinc. We can clearly observe that we have taken different synthesis time, that is 1 hour, 2 hour, 3 hour, and 5 hour, and 4 hour, to check which material is showing better CO2 absorption performance. And we observe that if we see the 1 hour sample, there is no particle size is very not crystal structure not formed properly. And if we see as reaction time increase from 1 hour to up to 5 hours, the particle size start increasing. This is a similar, SEM image of UTSA-16 cobalt that we already first reported by microwave method. This is a particle size and SEM image of UTSA-16 cobalt made by a solothermal method. If we compare the particle size of this solothermal as well as microwave method, we clearly observe that the particles of UTSA-16 cobalt made by a microwave method shows a bigger particle as compared to UTSA-16 cobalt made by a microwave method. See, UTSA-16 4-hour shows a very small particles. These are aggregate with each other. That's why it's showing little bit big particle. But if we see a single particle which is aggregated in one big particle, we clearly observe that it is very small as compared to a solothermal synthesis method of UTSA-16. So another one of the good application and characterization in this my research work is surface area and total pore volume. If we compare these two MOFs, that is UTSA-16 cobalt and UTSA-16 zinc, we observe that the surface area of each material at different synthesis time increases as we increase a reaction time. And if we see the 4 hours sample shows a maximum surface area as well as maximum pore volume. And if we increase a synthesis time beyond 4 hours, we observe that surface area as well as total pore volume start decreasing. Similar thing observed in case of UTSA-16 zinc as well. It shows high surface area as synthesis time increase and shows a maximum surface area at 4 hour synthesis time. But if we compare these two MOFs at 4 hours, UTSA-16 cobalt shows a little bit high surface area as compared to UTSA-16 zinc. But UTSA-16 zinc shows a high total pore volume as compared to UTSA-16 cobalt. So general property of MOF is that it is necessary to capture CO2 from flue gas. So flue gas contain humid air as well as SO2 and NO2. So our most important application of MOF is CO2 capture. So it is necessary that the MOF is stable under humid as well as SO2 and NO2 environment. So to check the stability of our MOF under this humid SO2 and NO2 environment, we perform a stability test. That is, we exposed our new made MOF, that is UTSA-16 cobalt and UTSA-16 zinc under humid as well as SO2 and NO2 environment. So this is chemicals that we used, NaHSO3 we used to generate 15 ppm SO2, as well as NaNO2 we used to generate 10 ppm NO2. And we kept a particular pH, that is for SO2 generate, we kept a pH of aqueous solution 3.7, and for NO2 generation, we kept pH 3 for NO2 generation. And also we used airflow to transfer generated SO2 or NO2 towards the desiccator, which contain exposed MOF samples. So this is the result after exposing our MOFs to humid as well as SO2 and NO2 gases. If we see the first MOF that we made UTSA-16 cobalt, this is our first achievement, that we reduced time up to 4 hours. So we can see that after this is black bar represent before exposure. This bar represent after NO2 exposure, and next one is after humid and after SO2. We clearly see that this material is stable under NO2 environment. But if we expose this material to humid, this CO2 absorption capacity slightly decreased as compared to previous one, as well as after SO2 exposure, I mean, this capacity decreased up to 40%. So the performance of UTS-16 cobalt is better only under NO2 exposure, but it decreased by around 27 and 40% after exposing to humid as well as SO2 exposure. So our basic need is that the developed material should show a stability under humid as well as SO2 and NO2. To fulfill these requirements, we developed this UTS-16 cobalt, and we clearly observe the result that it shows a better stability after exposing to humid. That is, CO2 absorption capacity decreased only around 3 to 4, as well as after SO2 exposure, it shows around CO2 capacity decreased around 8%.
Sorry for interruption, Sanjit. Can you conclude? Sorry.
Yeah. Just two slides remaining. And after SO2 exposure, it just reduced to 10%. This is a XRD of our lab made UTS-16 and UTS-16 cobalt. So this is our selectivity. It is necessary that to capture CO2 from NO2, it has to show a maximum selectivity. So this UTS-16 cobalt shows higher selectivity around 104. Also UTS-16 cobalt zinc shows a 118 CO2 selectivity that I mentioned here at 298 Kelvin and 1 bar. This is our conclusion that first we developed UTS-16 cobalt by microwave method by reducing time from 48 hours to 4 hours. Then we checked the stability of our developed material, but it's stable, that our developed material is stable for NO2 only, but unstable, comparatively unstable for humid as well as SO2. So we developed a new MOF, that is UTS-16 zinc, by replacing cobalt, and it shows a better performance under humid as well as SOx and NOx exposure. So this is my conclusion. Thank you, ma'am, for giving me opportunity to present my research work here. Thanks a lot.
And nice work, Sanjit. Nice presentation too.
Thank you for your kind attention.
Thank
you, ma'am.
Zinc is the better one, right?
Yeah, zinc is better.
Yeah, because it is the second one which are done after cobalt.
Yeah, zinc is better. If we see the capacity wise, I will show you. Just a minute. If we see the cobalt has a maximum capacity, but around 4.5 millimole. But if we expose to SO2, humid and NO2, its capacity is decreased. I forgot to mention exposing time. That is 2 days we exposed this sample. But if we see the zinc, it shows consistent performance after exposing to SOx, NOx and humid air.
You think your UTS-16 zinc is applicable to the world?
Yeah.
That's good. No other, anybody has any questions for Sanjit? Nobody has posted questions though. Anybody has questions? Can
Ma'am, shall I proceed for the next presenter? Dr. Pooja, ma'am.
Sanjit.
Yeah, I'm closing now.
Dr. Pooja, ma'am. After Pooja, ma'am, Lakshmi, ma'am, and Suryakala, ma'am. Anyone can present. Dr. Pooja, ma'am.
Yes, ma'am, I'm here.
Okay, ma'am, I request you to conclude your talk by 5 to 6 minutes.
Yeah, definitely, I will do so.
Yeah. At that time, our video will open so that you can conclude, please. I request. Yeah.
Okay. Yeah, so should I, is my screen visible?
Yeah. No, ma'am. You can start.
Okay.
share the screen, ma'am.
I'll share my screen first. Okay, so I'm sharing my screen. That is my Is it visible, ma'am?
Yes,
it's visible. Go ahead, ma'am.
Okay. So this is the work with my one of PG student Anil Govekar in his dissertation program, and I'm Dr. Pooja Jagesha along with my colleague Mary Stephen and Anil, we have worked on qualitative screening of phytochemicals present in curcuma longa, turmeric powder, with the different species of turmeric. Now, why we thought of doing of this work? Because nowadays we are seeing the turmeric is used in many of the products. You know, in the cold shakes, we have turmeric latte, then turmeric tea, which is now coming up very recently. Turmeric coffee is also available. So we just were curious to know that what should be the specific proper method to prepare the basic infusion for any products, any shakes which we are using to use the turmeric, and how to get the maximum antioxidants being extracted from the turmeric into the drinks which we are consuming to have a maximum effect on, to have a maximum antioxidant properties, we can take those. Now, as we all know that medicinal plants, and even the WHO has said that medicinal plants are the best source to obtain a variety of the drugs, and turmeric has been traditionally used in Asian countries as a medical herb due to its antioxidant, anti-inflammatory, antimicrobial, and anti-cancer properties, and is a potential against various malignant diseases also. And the objective of this study was to evaluate the antioxidant activity of curcuma longa. And what we did, we prepared different methods were used to prepare the turmeric tea infusion by using simply water as a solvent. Though the turmeric shows lots of antimicrobial activities in ethanol, but it didn't show antimicrobial activity in water. But main our aim was to prepare the turmeric tea, basic infusion for turmeric tea. And 4 different type of infusion were prepared, which I'll slowly go and tell in my PPT. Now, as we know, apart from knowing the various phytochemical properties of turmeric and the various antioxidant it has, we had used different methods from the Satara, Kerala, Mumbai region, organic haldi, which I got from the Nature Basket, and we found out in all the different turmeric varieties, we screened out the phytochemicals present in all these species, in all these turmeric variety. Now, also, we know that tannin and flavonoids are the most antioxidants which are present, which are very important antioxidant. And in this study, we tried to carry out the quantitative estimation of flavonoid, but just by the known method. We do have a proposed method, but due to COVID pandemic, we couldn't carry out our study. So today, I'm just giving you the results of the known methods. And for the proposed method, which is under research still, we have optimized condition but not validated our method. So that proposed method I'm not showing in my presentation today. So the very important thing what we did is, by using the different turmeric samples, we carried out different methods of preparation of turmeric tea. This one was the soft infusion, hard infusion, ambient infusion, cold infusion. In the soft infusion, as you can see in the preparation method, what we did is we weighed for carrying out the quantitative analysis, we weighed the turmeric, and here also, yeah, approximately 2 gram of turmeric was taken, and we added in 20 ml of water. And when we were analyzing more turmeric, then accordingly we were increasing the amount of the water. Now, for soft infusion, what we did, turmeric was blended in warm water, having the temperature of 70 to 80 degrees centigrade, and just it was kept for 3 to 5 minutes. Now, here, this method is used by the Chinese people for the tea preparation method. And now, in the hard infusion, in this brewing method, turmeric was infused in distilled water, having the temperature of same 70 to 80, but here it was kept for 25 to 30 minutes almost. And in ambient infusion, this method involves the mixing of the turmeric with distilled water at room temperature. Though we kept at the room temperature for 30 to 40 minutes for the ambient infusion. And cold infusion was used, where turmeric was infused in distilled water, maintained at room temperature for 15 minutes. And then this was refrigerated for an hour. And then I'll just... these are the results we got. By using different samples, all the infusions were prepared, and qualitative analysis of phytochemicals were done in different turmeric tea infusions. Sorry, there's a typo error. It's
yeah, that's okay.
Yeah, it's a alkaloid, flavonoid. These are the different tastes how we carried out I'm not describing the taste. These are the known tastes for flavonoids, xanthoprotic steroids, tannins, saponin, ferric chloride taste, terpenoids, glycosides, reducing sugar, resins, balsams, anthraquinone, chalcone, anthocyanin, coumarin, imodins. All these phytochemicals were screened in each and every sample. And then in the quantity, this is a method which I told you I've written again. Now I'll show you my results also later on, but what the conclusion that antioxidant properties and antioxidant effect of three types of turmeric as per these method was successfully screened. And among all these brewing method, what we observe, what hard infusion shows the higher antioxidant potential as compared to soft and ambient infusion. And so what we can conclude that whenever we are preparing, any of the samples of the turmeric showing that it has a turmeric and it's good for our health, then we have to see the basic infusion. It should be heated for approximately 25 to 35 minutes, and also depending upon the quality of the turmeric we are using. That also plays an important part here. Now here, these are the results. I'll just take one more minute. Flavonoid estimation, which was done by the known method. And determination of total flavonoid by use by quercetin as a standard method, and flavonoid content were measured as a quercetin equivalent. Calibration curve was prepared. And I'll now go to the results directly as I'm running due to time. These are the results which are showing in soft infusion, hard, ambient, and cold infusion. Cold infusion may very few antioxidants were present, and so it is advisable not to directly use the turmeric and in the cold extract. And these are the results of total flavonoid present in the different tea infusions, ambient, soft, hard. So quantitatively also it was observed that the hard infusion where we boiled it for 25 to 30 minutes is showing the maximum amount of the flavonoids. This is by the known methods. Proposed method has been done by using boric acid, and as we are under the process that I am not reporting here, as I told you, this is the calibration curve for flavonoids. And these are the reference works, references I've used. And also we have done the microbial study, but in water, the microbial zone we could not found out, but with ethanol solvent, the turmeric, these extracts are showing the microbial activity. My student Anil just would like to tell you about, for 30 seconds, what he's doing in his future project. Anil, are you there?
Yes, ma'am.
Yeah.
Hello, everyone. I'm a post student of Vivekananda Education Societies, College of Science and Commerce. In my dissertation work, we worked on quality analysis of phytochemicals. We selected some turmeric samples. In that, we analyzed quality analysis of phytochemicals, and we worked some quantitative studies for flavonoids. And due to pandemic, we not able to study more flavonoids. And after that, we are also planning for antimicrobial studies of some flavonoids. Due to this pandemic, we also know that these phytochemicals are mostly used to cure our disease, like normal fever. It will help most useful. So thank you, and we are looking forward for more better results. Thank you, ma'am.
Yeah, thank you all, and I hope I have not taken more time.
Nice presentation. Only thing I have a query is that basically you said microbial activity is not seen in water. That was the thing.
Yeah, that we are confirming, ma'am, because we could not see any microbial zone. We tried the different cultures. I have those results also, but then it was then shown in ethanol as a solvent. So we are again making our infusion more concentrated one to check whether we might get the antimicrobial activity of
because
those.
we cook curcumin in water only, so.
Yeah. So because we use water, so our main emphasis is to see everything with the water as a solvent.
Yeah. Nice work, ma'am. All the best for future.
Thank you so much, ma'am.
Thank you, madam. I'd like to thank all the participants for this 3 to 4 members for the first session and chairing session by Dr. Annapurna ma'am and Domal Ramesh sir. Thank you very much, ma'am. And the second session, I would like to invite chair as Dr. Rupa ma'am and Dr. Domal Ramesh sir to chair the session for 3 to 4 members of oral presentation. I request Rupa ma'am and Domal Ramesh sir, please welcome to the session. And now I request Lakshmi ma'am and Suryakala ma'am.
Okay, ma'am.
Lakshmi ma'am and Suryakala ma'am from Vizag.
Yes, madam. Yes, we are available.
Yeah, ma'am.
Shall I share screen, madam?
Yes, you can. Ma'am, time will be 5 to 6 minutes.
Okay, madam. Okay. Is it visible, madam?
Just started.
First of all, belated Teachers' Day to all the teachers here. Next, I would like to congratulate Dr.Y.Prashanthi madam and her team for conducting this well-organized webinar. It's very nice, madam, and thank you for giving us opportunity for present our work. And I am Dr. Suryakala from Geetham Institute of Science, Geetham University, Vizag, and this is my team. And our work is adsorption of rare earth elements, praseodymium and gadolinium by nano cobalt oxide, and adsorption isotherms and kinetics. So the abstract I would like to share with all. So nano metal oxide, that is nothing but CO3O4, is prepared efficiently in a simple, eco-friendly and cost-effective manner. Thermal decomposition of metal oxalates which lead to metal oxides is considered to be a versatile method for the synthesis of transition metal oxide. So various characterization techniques like XRD, FTIR, SEM are applied to exposure to explore the morphology, bonding nature, size of the nanoparticles synthesized. The adsorption of rare earth elements, especially praseodymium and gadolinium onto synthesized nano cobalt oxide, is further studied using ICP-AES method. The influencing parameters such as adsorption efficiency, which include contact time, pH, initial concentration, and temperature are studied. The adsorption isotherms, kinetics, and thermodynamic parameters are analyzed. It is observed that adsorption studies follow pseudo second order kinetics and follow adsorption isotherms of Langmuir. So coming to the introduction, the surface area of crystalline materials increase with decrease in particle size, which increases the adsorption sites on the material. Among all the cobalt oxide, the most stable phase is a p-type semiconductor with high Young's modulus, which varies between 115 to 160. The nano powder of cobalt oxide is extensively applied in a gas sensor, magnetic catalysis, lithium-ion batteries, etc. So the present work we propose a simple, eco-friendly and most effective method for the metal oxalate preparation, and from which by using thermal decomposition method, we have prepared metal oxide. So several methods, that is chemical precipitation, ion exchange, electrode deposition, various membrane separation, etc. Various methods are available, but still we depend on this thermal decomposition method as it is eco-friendly and simply pouring one solution into another one without disturbing the environment. Then coming to ICP-AES, so this is inductive coupled plasma atomic adsorption spectroscopy, so which is used for the adsorption studies, and CO3O4 synthesized through thermal decomposition method is used as adsorbent. And the rare earth elements we have chosen are praseodymium and gadolinium. Next, coming to experimental details, cobalt nitrate hexahydrate, CO3 NO3 tries .6H2O, and sodium oxalate and gadolinium nitrate are used, and all the chemical reagents are commercial and are of analytical grade, and the stock solutions are prepared from reagent grade compounds using double distilled water. Coming to synthesis, the synthesized metal oxide CO3O4 is characterized by using various analytical tools such as XRD, SEM, FTIR, BET, etc. So the various functional groups and elemental composition, material characterization, and surface area, the information can be obtained from it. So the SEM image of this can be observed here. Then coming to XRD and FTIR. So this is the XRD of the synthesized CO3O4. and the FTIR of CO3O4 is this. The BET analysis was employed for exploring the surface area and the porosity. So here we obtained nano cobalt oxide is measured to be 88.37 units. Coming to batch adsorption studies, the important objective of this work is to absorb rare earth elements such as praseodymium and gadolinium from aqueous solutions using nano cobalt oxide by the ICP-AES technique. So these, as we discussed, so the parameters like initial dye concentration, adsorbent dose, contact time, and solution pH, temperature, etc. are studied. Coming to effect of pH, see, you can see on this graph, so the pH 6.5 is obeyed for both the rare earth elements, and the contact time is observed to be the 40 minutes for this, and this can be observed in the graph. So next, effect of adsorbent mass. So we have started from 5 milligrams to 20 milligrams of adsorbent, but we have observed to be 10 milligrams of the cobalt oxide is appropriate for this adsorption studies. Coming to the next parameter, that is influence of initial dye concentration. So dye also we have varied its concentration from 1 ppm to 30 ppm for both rare earth elements. So these are the findings we observed. Then effect of adsorbent mass. So coming to Langmuir adsorption isotherm. So this isotherm explains the adsorption of appropriate homogeneous phases, and it is specific on the surface of adsorbent, which is observed to be monolayer. So the linear Langmuir adsorption isotherm model is like this for both the rare earth elements. Then adsorption kinetics. So adsorption is the processor which normally quick and instead of inner and outer dissemination. So this adsorption studies we have conducted at various temperatures starting from 293 Kelvin to 323. You can observe the graph. Then these are the parameters what we have concluded from the obtained graphs. So next, coming to thermodynamic studies. So the delta H values for both rare earth elements are these. This is delta S, that is entropy, and Gibbs free energy. So these are the values we have obtained from these graphs. And coming to conclusions, so from the above results and discussion, it is understood that nano cobalt oxide is successfully prepared and characterized, and it is used as adsorbent for both the rare earth elements, and ICP-AES technique is excellently applied on these, and the parameters like pH, initial concentration, etc. are studied. So from these studies, it is extended that the feasibility of the reaction is observed, and also the pseudo second order kinetics best fit into the model. And it is concluded that this nano cobalt oxide can be used as better adsorbent for further rare earth elements also. Our future scope of the work also includes other rare earth elements, like we are studying on lanthanum and neodymium, etc. So our study is to study the various adsorption studies by using this adsorbent. Okay, madam. Thank you.
Okay, ma'am. Wonderful presentation. Your expression is effective manner and systematically delivered. Congratulations. Next, over to Prashanthi ma'am.
Thank you, sir.
Thank you, Pooja ma'am, and thank you, Ramesh sir and Rupa ma'am. And the next speaker,
Madam, I have one doubt.
Yeah, ma'am, please. Over to Rupa ma'am.
Madam Suryakala madam, among these praseodymium and gadolinium, which is the best rare earth metal?
Gadolinium only we got results better manner, madam.
Okay, thank you. It's a nice presentation, madam. Thank you.
Thanks, madam. Thank you.
Next, I would like to invite Dr. Ramarao Garu from Kakinada. Sir, you can conclude your talk by 5 minutes. Dr. Ramarao Garu. Sir, Ramarao sir. Ramarao Garu. Otherwise, Abhinaya madam. Abhinaya. Next, Mr. Hari Prasad. Otherwise, next, Mr. Ambati Chandrasekhar from US. Among these four who are ready, please come forward to present your oral presentation by 5 minutes.
Madam, good morning, good afternoon, madam. So
Good
myself,
afternoon.
Hari-Prashanthi.
Yeah. Your voice is not audible. Please increase your volume.
Okay, sure, madam, sure.
I request next participants be ready with their presentations. Dr. Ramarao Garu, Mrs. Abhinaya, and Mr. Chandrasekhar sir from US.
Okay. So good afternoon to all respected eminent speakers for this international webinar on frontier in science and technology, 5th and 6th September 2020. So harmful thanks to all the Department of Chemistry and Pharmaceutical Sciences. Ma'am, Prashanthi ma'am, my voice is audible to you, ma'am. Prashanthi ma'am.
It is not, sir.
Prashanthi ma'am.
Hari-Prashanthi sir, it is not audible and visible also. Now it's okay.
Is audible to you, ma'am?
Now it's okay, sir.
Ma'am, so here my topic is synthesis and application of metal organic frameworks. So myself, my name is Hari-Prasad under the supervision of Prashanthi ma'am, so Department of Chemistry and Pharmaceutical Sciences. So here the introduction of my topic is metal organic frameworks, madam. So here over the last 2 decades, the metal organic frameworks have received extensive interest due to their structure related properties and endless number of possible structures. The MOF are well studied for their potential storage and separation, catalysis, luminescence, molecular magnetism, and various other properties. In this metal organic frameworks, with the luminescent property, have attracted considerable attention in sensing the organic molecules, and the MOF, the dynamic behavior, and the guest responsive transformation of the MOF made it unique candidates for the detection of pollutant, explosive, and biological molecules. The luminescent, my topic here, this is luminescent metal organic framework. So made by using a flexible ligands to synthesize soft and guest response metal organic frameworks. The probes in detection of organic molecules at ultra trace levels have been more focused. So unlike rigid metal organic frameworks, the conformational orientations according to the size of the guest entity are adjustable by flexible metal organic frameworks. Okay, ma'am. So here, this is luminescent metal organic frameworks for the detection of the luminescent property of various nitroaromatic compounds are important in addressing the global issues like pollution control and presently security control. These nitroaromatic compounds are derivatives or reactants of dye, pesticides, pharmaceutical industries, and the molecules such as 2,4,6 trinitroenylene, trinitrotoluene, nitrophenol, 4-nitrophenol, and 2,4,6 trinitrophenol are used for making explosives, and these molecules are considered as health hazards. Even in a trace quantity, here in our work is aimed to synthesize the novel cobalt.
Hari-Prasad sir, it is not audible. Make it clear.
Okay, madam. Luminescent marks that are effective for the detection of nitroaromatic compounds at ultra trace level. So metal organic frameworks with flexible ligands, madam. Hello, madam. Hello, madam. Okay, madam. Okay, madam. Thank you, madam. Thank you, Rupa, madam. Ligands with free carbon-carbon rotation for conformational freedom are been focused. So here I have synthesized the 3 metal organic framework compounds by using the flexible ligands BPEB and HFI-PBB and so OBA ligands, so deoxybenzoic acid. So here by the synthesis, novel synthesis method. So these are the structure of compound 1. So this is it is a distorted octahedral geometry. So this is the BPEV ligand, and so these are the HFBAB ligands are coordinated to the cobalt metal atom. So here the blue color is the central cobalt atom, and the red color is oxygen, and indigo blue is shows nitrogen atom. So this is the structure of secondary building block unit with the coordinating molecules of BPEB and HFBIB ligand. So this is the compound 1 related information, madam. So this is the compound 2. So this is the zinc metal organic framework. So by using the same ligands BPEB and HFI PBB. So here it is a distorted octahedral manner only. So this is the lost by cobalt metal atom, but here we have using different organic linker. So that is DVA and with BPEB ligand. So here these are the structures we get from single X-ray diffraction technique, and also we have done thermogravimetric analysis, and this is the luminescence work we have studied. This is the three compounds, cobalt and zinc and cobalt 2. So these are the compounds. This is the different absorptions of by using various nitro compounds, dinitrobenzene 1, 2 dinitrobenzene 1, 3 dinitrobenzene, chlorodinitrobenzene 2, 4 dinitrophenylhydrazine, and 4 nitroaniline, and 4 nitrophenol. So these are the so most of the studies these three compounds showing especially the absorptions are I naught by I value of 4 nitrophenol and nitroaniline can showing the better graph. And the next here this is the quenching studies of these three compound. This is the compound 1 quenching study, and this is the compound 2 quenching study zinc, and this is the compound 3 quenching studies. So here the quenching studies also gave the evidence for these compounds. Nitroaniline and nitrophenol can shows the maximum quenching up to 85 to 90% region. So all these three compounds, and our further studies for the future further future studies, having the idea we want to explore these metal organic frameworks as a photosensitizer to activate small molecules, and these metal organic frameworks can interact specifically with the nitro group of the organic molecule. So they can be used as a photocatalytic degradation of nitro group containing the organic dyes. So thanking for the giving opportunity, ma'am, all the Department of Chemistry. Thank you, ma'am.
Hariprasad, sir.
Ma'am.
You have said that nitrophenol is a good work. It is working as a good quencher.
Yes, ma'am.
Have you checked with any nitro group being an electron withdrawing group? It might be working as a good quencher.
Yes, ma'am.
Have you checked with an electron releasing group phenols?
Yes.
Are you getting?
Hello.
Okay. Okay. Thank you, Hariprasad, sir.
Okay. Okay.
Nice presentation. It is. Okay. Go.
Okay, Hariprasad, nice presentation.
Thank you, ma'am. Thank you, sir. Now I request Dr. Ramarao, sir, from Kakinada. Sir, please, I request you to conclude your talk for 5 minutes. Dr. Ramarao, sir. Dr. Ramarao garu.
Sir, madam, okay, yes, sir. I am sharing my slides.
Yes. Please, you can, for 5 minutes, sir.
Okay, okay.
5 to 6.
Madam Prashanthi, my presentation is visible?
No, sir, not yet started. Ramarao garu, not yet started.
No, okay. Okay, madam.
No, sir. Not yet. I request next participant be ready. Ramarao garu, you can share the screen. Not yet started.
Okay, I am overall explain, madam. This is
Okay.
some.
Okay, you can.
Okay. Thank you, Prashanthi garu, your dynamic leadership. This seminar is very wonderful. My classmate Prashanthi, congratulations. I D. Ramarao, Dr. Ramarao, Kakinada. My presentation is applications of nanomaterials in various fields. There is no, we all know about what is the nanomaterials. It is the study of extremely small structures having size of the 0.1 to 100 nanometers. This is the very applications in various fields. That is in wonderful applications in the medical field, drug delivery system. This is also very wonderful subject. The energy and the environment is discussed detail applications. The nanoparticles in drug delivery, the drug delivery system and the peptide delivery, cancer treatment, and also the applications of the various nano systems in cancer therapy, such as carbon nanotubes and nano crystals, nano wires, nano tubes. In the present days, the nano medicine technology, this is also very wonderful uses. Nano robots also available. The operation, the wonderful techniques in present situation. The application, the main applications, the different fields in nano materials, health and medicine, electronics, transport and energy and environment, space, and also industry applications in the food safety measurement and chemical industry. The wonderful applications there in the nowadays, the nano materials are wonderful uses. The particle size less, the wonderful uses is there. In the future days, there is also the wonderful applications developed day by day. It is the nanotechnology, the growing subject. That's why the applications is wide range of applications in drug delivery system also. In nanotechnology, nanoparticles are used for the site-specific drug delivery. In these techniques, the required drug dose is used and side effects are lower significantly as the active agents in deposited in the motion. So nano technology and energy and environment. Nanotechnology will play the critical role in coming 50 years by the protecting of the environment and providing sufficient energy for the growing world. And also applications of nanoparticles in drug delivery. This is the nanoparticles are used to deliver the drug with enhanced effectiveness of the treatment of the head and the neck cancer, neck and cancer. And also the final we conclude the nanomaterials have increased surface area and nanoscale effect, hence used as a providing tools for the advantages of drug and gen delivery, biomedical and immigrations and the disintegrated biomass nanomaterials having unique physiochemical and biological process in the future and present days. The technology by it meaning with other technologies and the very wonderful subsequent energies of complex and innovative hybrid technologies, biology based techniques also it is developed in the future days. The ability of nanotechnology to engineering matters and the smallest scale is revelations are such as information technology, connective connective science and biotechnology, and is leading to the new and interlinked, there are other fields. Thank you very much, Prashanthi garu. Wonderful. I am unable to slide sharing. There is a very less time. Anyhow, I will. Thanks to the organizers for giving a few minutes opportunity. Thank you.
Thank you, sir. It's a good presentation.
Thank you.
Now, I request the last participant, Mr. Ambati Chandrasekhar Garu from US. Sir, are you available, sir?
Yes, ma'am.
Yes, sir.
Yes, is it audible to you?
Yeah, voice is okay. You can start sharing, sir.
Sure, ma'am.
I request you to conclude your talk by 5 to 6 minutes. Sorry for inconvenience.
Sure, ma'am. So is it visible to you?
Yes.
Okay, so here I would. Yeah, so here I'm going by slide. So I would like to present a topic on identification and quantitation of malonic acid biomarkers of inborn errors of metabolism. So it is an analytical method where we developed by derivatization procedure. So the contents include the introduction, objective, methods, results, and conclusion. So in introduction, I would like to say that inborn errors of metabolism constitutes a wide variety of genetic disorders, including malonyl coenzyme A decarboxylase deficiency, vitamin B12 deficiency, and ethyl malonic encephalopathy, and they have been reported to have serious clinical consequences. So means the malonic acid, methyl malonic acid, and ethyl malonic acid are the three carboxylic acids. They play a role in genetic disorders, different genetic disorders. Okay, so so they lead to the deficiency of these enzymes leads to the different disease conditions. So what was the objective is to develop a robust method for the determination of malonic acid, methyl malonic acid, and ethyl malonic acid by LCMS in a single method, and apply this method to the different biological samples like plasma or serum or cell pellets, anything. And this is the methodology. This is a schematic representation of the reaction where we derivatized the malonic acid, methyl malonic acid, ethyl malonic acid with the 3-metanitrophenyl hydrogen. So when 3-metanitrophenyl hydrogen interacts with the dicarboxylic acids, it converts into hydrogen derivatives, where we optimized the reaction conditions. So the solvents like how much percentage of acetonitrile we have to use and the temperature, time, we have optimized. In the optimized conditions, we have used 50% of acetonitrile and incubation for 30 minutes of time at 40 degrees temperature. And so for this derivation method, we have also spiked isotopic labeled standards like malonic acid 13C2, methyl malonic acid D3, and ethyl malonic acid D3. And mobile phases used for this method were 0.1 formic acid in water and 0.1 formic acid in acetonitrile. The column used is acuity UPLC, water's acuity UPLC, C18 column we used, and the experiment was performed on Agilent 6495 LCMS triple quadruple LCMS system. So this is the extraction procedure for the malonic acid, methyl malonic acid, and ethyl malonic acid. So we have to derivatize in this reaction. So you have to take equal amounts of like standards and the biological sample like plasma, take 40 microliters, and you have to spike the 5 microliters of isotopic labeled internal standard mix, and after that add 20 microliters of freshly prepared 200 millimolar metanitrophenol hydrogen and 20 microliters of freshly prepared 120 millimolar ethylene diamine chloride. And where these mixture is to be incubated for 30 minutes and subsequently cooled on ice for 1 minute. Then reaction mixes were diluted with 1.91 ml of 10% aqueous acetonitrile and analyzed by LCMS in MRM mode. multiple reaction monitoring mode. So these are the results in this diagram. You can see the three malonic acid, methyl malonic acid, ethyl malonic acid, all three were separated nicely in a chromatogram. And you can see the in each chromatogram, you can see, I can see just a pointer options I am showing. See here, you can see the internal standard spiked into the malonic acid, and internal standard spiked into methyl malonic acid, and internal standard spiked with the ethyl malonic acid. That is the EMA D3, methyl malonic acid D3, and malonic acid 13C2. These are isotopic labeled compound along with the malonic acid, methyl malonic acid, and ethyl malonic acid. They are eluted at the same retention time. You can see both three peaks. So advantages of this method is great peak resolution and great peak shape, increased sensitivity, increased quantitation accuracy, no matrix effect, and longer stability of the derivatized sample. So if the physiological concentration of the sample is very low in the if the sample concentration is very low, also you can test by this method. And the same, these are the chromatograms from the samples. We have used two types of plasma from wild type plasma, and another one is malonic acid carboxylase deficiency mice. So here you can see the elution of malonic acid and malonic acid 13C2 at the same retention time, and malonic acid, methyl malonic acid D3, and ethyl malonic acid and ethyl malonic acid D3. So you can see the nice chromatograms and high accuracy and quantitation. Conclusion is advantage of malonic acid derivatization using metanitrophenyl hydrogen is it greatly enhances the detection sensitivity as well as increased quantitation accuracy, and good linearity and precision were obtained with the quantitative determination of target molecules. So I can say this is an unambiguously identify and quantitate target molecules in submicromolar and nanomolar range concentrations. This is a well efficient developed methods. Thank you. If you have any questions, please let me know.
Yeah, Chandra, it's a very nice presentation and a great job
Thank
you
you.
have done, and congratulations.
Thank you so much, sir.
Yeah.
Thank you so much for the Dr.Y.Prashanthi madam for giving me the opportunity, and all the coordinators of this event.
It's a nice presentation. And
thank you, madam.
Thanks.
And one more request for 3 minutes, please. One ma'am, Mrs. Abhinaya ma'am from Coimbatore. I request Abhinaya ma'am, please conclude your topic by 3 minutes, because it's too late and I'm extremely sorry for all the participants. Please bear with us for 3 minutes.
Okay, ma'am.
3 minutes, please.
Okay, ma'am. Okay, ma'am. Hello, ma'am. My audio is audible. Hello, ma'am.
Yeah, audio is okay, ma'am. Video is
My screen?
about to start.
Yes, ma'am. My screen is visible?
No. Just about to start. Yes.
Oh, okay, ma'am. Good noon, all. I am Abhinaya from Internationl University, Coimbatore, and I am doing my PhD work under the guidance of Dr. Y.Prashanthi. I am going to explain the synthesis, characterization, and molecular docking of N-aryl amide pyridopyrimidine 2-ons. And we know that nitrogen heterocycle compounds are more important in the drug discovery, in which pyridopyrimidine plays an important role because of the valuable biological property. So our objective is to prepare the pyridopyrimidine ylidine acetic acid and convert it to amides using thionyl chloride. And we characterized using IR, proton NMR, C13 NMR, and mass spectrum. And we studied the molecular docking interaction of all the compounds. And general preparation is we prepared the pyridopyrimidine ylidine acetic acid, and it is treated with thionyl chloride at room temperature, and it converted as acid chloride, and that acid chloride is again treated with various amine at 0 degrees Celsius to get the amides. And the molecular docking for all the synthesized compounds were carried out against the active site of 1T9U and 3UZ0 protein from the E. coli and Bacillus subtilis bacteria. using Autodoc 4.2 software. These are the acids and the amines we use to prepare the amides. And the resultant discussion, that is, we prepared the pyridopyrimidine ylidine acetic acid, and then this acetic acid is treated with thionyl chloride at room temperature, and again converted as acid chloride. Then the acid chloride is reacted with reacted with various amines like naphthalamine, aniline, para-toluidine, and para-anizidine to give the respective amides. These are the eight compounds we have synthesized in our laboratory, and we also carried out the characterization for all the 8 compounds. So here I am explaining for only one compound. That is, this is the proton NMR of the acid with aniline. That is that amide here, that 4.7 and 6.5 so shows the presence of the peak shows the presence of hydrogen outside the ring, and the 7.0 to 8.0 the multiple peaks shows the presence of the pyridine ring as well as the benzene ring. And we also carried out the C13 NMR, in which it gives 16 carbon peaks, which can also confirm the structure of the synthesized amide. And we also taken the molecular mass, that is the mass spectrum, in which the molecular ion peak is 281. So from this all characterization, we confirm the structure of the prepared all 8 synthesized amides. And we carried out the molecular docking for the synthesized compound for 8 compound also. So using these 2 particular proteins, in which the compound A, that is the compound acid with the naphthalene amine, shows higher binding energy, that is minus 10.4 and minus 10.7. So the compounds, the naphthalene, pyridopyrimidine, ylidine acetic amide is the highest binding energy with these 2 proteins, and the binding energy is minus 10.7 and minus 10.4. And we also calculated the van der Waals forces and hydrogen bond of interactions, and the energy also calculated, the pi bonding, pi interactions also calculated. This is one of the image, that is a molecular docking image of our synthesized compound with the protein 1T9U. So this comparison table. So the summary is we synthesized 8 pyridopyrimidine amides and confirmed structure by following characterization, and the molecular docking revealed that the naphthalene pyridopyrimidine acetamide is a high binding energy. So based on the docking results, we claim that these compounds could serve as an effective antimicrobial compound to treat the bacterial infection. So these are the reference, and thank you.
Yeah. Vinay ma'am, congratulations.
Thank you,
okay,
sir.
Wonderful presentation. But one question.
Yes,
you have shown the C13 NMR.
Yes, sir.
The bottom line is having a lot of noise. And yeah.
Yes, sir.
Yeah. Then, and one more thing is how you are removing the acetic acid traces. Can you explain?
The acetic, that is by dissolving that prepared solid particle and filter and dry and purification by column chromatography. We purified by the chromatography. Column, with the column, sir.
By doing column chromatography. Okay, nice, nice. Yeah. Okay, done.
Thank you.
Ma'am, can you explain you are using the thionyl chloride?
Sir, I am not audible.
You are using the thionyl chloride. It is solvent or what? Converting acid?
No, sir. I am not getting. Hello? Sir, your audible is not.
Right.
Yeah.
So what? You are converting acid to acid chloride, right? And then after coupling
Yeah. Then we are coupling with the amines and we are converting it to amide.
My question is only for that. You are using thionyl chloride. Is a solvent or what? Is catalytic amount?
Thionyl chloride is a solid.
No, it is not a solid. It is liquid.
Sorry, liquid.
Yeah, it is not solid. It is liquid.
Not. It's liquid.
because it's highly toxic. And
sir.
are
Highly
you using
toxic.
solvent or you using as such as a solvent, just thionyl chloride?
Sir, what's that? Sir?
Hello.
Yeah, using as solvent, sir.
So then after the complete reaction, how can remove this thionyl chloride and how to confirm? Ethylene is not there, because otherwise whatever the amine unreacted is going to formation of that salt, and that's why you have the number of this is the noise in that.
Yes, sir. Yes, sir.
So actually,
if you wash with sodium bicarbonate, it goes off. See, thionyl chloride, it removes off.
Yeah, it should be used
bicarbonate.
only
Yeah.
the
After getting
equivalent
the
required
product, you have to wash with
equivalents
sodium
only,
bicarbonate.
and not as a solvent system. Yeah. Okay. Done.
Okay.
Congratulations.
Thank you, sir.
Thank you for all the participants for giving oral presentation, and extremely sorry because it's a request from all the participants to give an opportunity to present. And I like to thank Dr. Rupa ma'am and Dr. Ramesh sir for chairing the session. And thank you, ma'am. Thank you so much. And it's a small announcement. Again, we will meet by 4:20 for the further session. Thank you once again. Please be there by 4:20. And I will send feedback link of this by today evening for both the sessions. Thank you very much. Thank you all. We will meet by 4:20.
Thank you, ma'am. Thank you.
Thank you. I am ending the session. Thank